Mr. Prashant Pradhan, can you hear me?
Yes, sir. Hello, sir. Can you hear me?
Yeah, we can hear you. Perhaps you can, I don't know, somehow the sound is a bit low. Maybe it is
Good morning, Jayantada.
because
Okay, sir. Just wait. Just wait.
Pastor Ray, your microphone is muted. Pastor Ray.
No, sir. Microphone is on. Just give me.
Yeah. Now it is okay, I believe.
yes, it
Yeah,
is.
So can you hear me?
Yes,
যাচ্ছে?
yes.
হ্যাঁ?
হ্যাঁ, পরিষ্কার শোনা যাচ্ছে জানতদা।
Good. না আমি জানি তোমার সাথে তোমার শোনা যাবে, মাইক লাগবে না। ও মাইক।
এখন মেদিনীপুরে প্রচন্ড বৃষ্টি হচ্ছে, ফলে আমি জানি না কি ম্যানেজ করব।
আমাদের এখানে সকাল থেকে ভীষণ বৃষ্টি।
আজকাল সব জায়গায় চলছে বৃষ্টি, আমাদের নেই, কলকাতা এখন শুকনো।
মানে এখানে 2 মাস পরে আজ বৃষ্টি হয়েছে।
It's raining cats and dogs, and I don't know, the speed is very poor here, very low. I don't know how shall I manage.
Now, are you in Vidyasagar University now or in your apartment? Joe?
I am in my apartment.
Oh, good. Then electricity will not be a problem. That is the main thing.
Yes, correct, sir.
Sir,
তোমরা
now can you hear me?
কিছু কাজ ছিল না সকালে, সেটা সেরে নাও, তারপর আরম্ভ করব। হ্যালো, নটা তো বেজে গেছে।
So we can start now.
রাইট, প্লিজ।
So, Mr. Prashant, please go ahead.
Yes, sir. Can you hear me right now?
Yeah. I don't know. Are we using headphone or something?
Yes, sir. Headphone.
Yeah, headphone. Can you remove your headphone and directly speak? Are we in a mobile or in a laptop?
In a laptop, sir.
Laptop. Could you please remove your headphone and then try if the volume increases?
Okay, sir. Now can you hear me?
It's better now. It's better.
Okay, sir. Okay, so good morning, everyone. It gives me immense pleasure to extend to you all a very warm welcome on behalf of Chirantan RASAYAN Sanstha and Midnapore City College, who have clapped all of us to today's webinar, Science Beyond Boundary, Invention, Discovery, Innovation and Society, RASAYAN 10. The COVID-19 pandemic has created the largest disruption of education systems in human history, affecting nearly 1.6 billion learners in more than 200 countries. Closures of schools, institutions, and other learning spaces have impacted more than 94% of the world's student population. This has brought far-reaching change in all aspects of our life. It has changed the way we do almost everything. There is significant hope that this is temporary. But while we continue to live under various lockdowns and restrictions, how can we carry out further research? Because the pandemic, ever since it started, has scuttled the pre-existing normalcy. Stress, tension, and confusion are palpable. Academicians are no exception to that. Academic interaction has also been hampered to a great extent due to this situation. So, the need of the hour is to innovate and implement alternative strategies. We can't sit back thinking when the situation will be perfect. As an instigator of that spirit, the organization Chirantan RASAYAN Sanstha, in association with Midnapore City College, has decided to host this international level webinar to motivate the young upcoming researchers to continue their quest in spite of the all odds. That is why today we have assembled via Google Meet platform with live coverage on YouTube. So now let us have the pleasure of listening to the enchanting prayer for initiating our webinar. I would like to request our vocal performer, Dr. Prithvideepa Sahu, faculty member of the Department of Chemistry, Vishwabharati University, Santiniketan, to perform the invocation. So, Dr. Sahu, please.
Yes, thank you. Thank you, Dr. Prashant Pradhan. Good morning to everyone. Today I'm trying Saraswati Vandana. As usually I do, I start with some mantras or chanting. Today also I'm trying. I'm not sure whether my video is... I'm turning on my video, but the camera is not properly working at all. But still I'm trying. Okay. घोर रूपे महारावे सर्वशत्रु भयंकरी भक्ते भो भरदे देवी त्राहि मां शरणागतम् सुरार्चिते देवी सिद्ध गंधर्वे सेविते जाड्या पाप हरे देवी त्राहि मां शरणागतम् जटाजूट समयाक्ते लोला जिहन्ता कारिणी द्रुत बुद्धि करे देवी त्राहि मां शरणागतम् देहो मोने सुदूर पानी हारिए फेली आपनारे देहो मोने सुदूर पानी हारिए फेली आपनारे गाনের সুরে আমার মুক্তি ঊর্ধ্বে ভাসে এই আকাশে আমার মুক্তি আলোয় আলোয় আমার মুক্তি ধুলায় ধুলায় ঘাসে ঘাসে এই আকাশে আমার মুক্তি আলোয় আলোয় এই আকাশে আমার মুক্তি আলোয় আলো। Thank you so much. Thank you.
Thanks a lot, ma'am, for your
Thank
wonderful.
you. Thank you.
Yes, yeah. For your wonderful and mesmerizing enchantment. Okay, so now I would like to request the president of the society, Chirantan Rasayan Sanstha, and also the convener of this webinar, Professor Brajagopal Bag, to kindly deliver the welcome address. Before he starts, let me briefly introduce Professor Bach. Professor Brajagopal Bach did his Masters of Science in Chemistry from the Indian Institute of Technology, Kharagpur, and got his PhD from the premier research institute, the Indian Institute of Science, Bangalore. Then he moved to Germany with the prestigious Alexander von Humboldt Fellowship and spent 4 years in the Ruhr University, Bochum, Germany. He has already produced 18 PhDs and published over 70 research papers in leading journals and delivered over 80 invited lectures in India and abroad, including invited lecture presentation in the symposium organized by IUPAC. Based upon his academic achievements, the Ministry of Human Resource and Development recognized him as the Leadership for Academicians program, LEAP, in the year 2019, jointly organized by IIACR Kolkata and Harvard University, USA. He is the founder president of CRC and he is also the editor-in-chief of the Society Journal. With this brief introduction, I request Professor Brajagopal Bach, sir, to say a few words about the society. Please, sir.
Good morning, everybody. I'll try to switch on my camera if possible. Can you hear me? Am I audible?
Yeah.
Okay. Thank you very much. Honorable Principal of Midnapore City College and also the co-host of RASAYAN 10, Professor Pradipto Chakraborty, former CRS medal awardees present here today as guests. Today's recipients of medals who will present their award lectures, Professor Shanku Mallik, Dr. N. Selvakumar, Dr. Abhijit Mandal, the chairpersons, Professor Jayanta Kumar Roy, Professor P. Jaishankar, Professor Prasenjit Mal, Professor Madhusudan Maji, Dr. Abhishek Ghosh, Dr. Jagannath Pal, and Mr. Prashant Pradhan, faculty members, research scholars, master and graduate students from various institutions present here today, ladies and gentlemen. It gives me an immense pleasure to welcome you all to today's webinar entitled Science Beyond Boundary, Invention, Discovery, Innovation and Society, RASAYAN 10. Let me tell a few words about our society, Chirantan RASAYAN Samstha. It was established in the year 2016 with the following objectives: to promote research and development in all branches of science for a better living, to organize seminars, workshops, training sessions, and similar activities for the promotion of research and development in all branches of science, and outreach activities, to collaborate with established institutions and other scientific societies in India and abroad, so as to fulfill the goals and to carry out activities that may assist in or be necessary for the fulfillment of the above objectives. Since the beginning, the society organizes symposia and webinars, and since the year 2017, we have organized already 9 symposia and webinars, and this is the 10th one. And also, the society honors eminent scientists and academicians by the presentation of CRS medals. And since the year 2017, we have already awarded 22 scientists and academicians, and this time we will be awarding 3 scientists. And as you can see here, we have awarded scientists and academicians from different countries from all over the world. And also the society has published one journal named Prayaging Rasayan. Its name in English is Applied Chemistry, and in German it is Angewandte Chemie. It is a quarterly open access peer-reviewed journal. Here the publication is free of charge, and also the articles are downloadable free of charge. It has got ISSN, and recently have become the member of PROSREF, means every article should have DOI and the articles are checked by authenticate as suffice to mention on science beyond boundary. And students are the future leaders of science, technology, and society. Keeping that in mind, the society organizes various programs for students and research scholars. So we have got the Students' Corner, poster presentations, student-scientist interaction, chemistry quiz, felicitation of school toppers, and also demonstration of food processing, preservation, and packaging as an outreach activity. So as you can see here, the students are felicitating or enjoying the award of this best poster in 2019, and here felicitation. And I was fortunate to take part in this Leadership for Academicians program, LEAP India 2019, which was organized jointly in association with Harvard University, US. So I visited Harvard University, and that time there was full of snow. And I was walking around, and this particular statue drew my attention. And on the other side of this, you can see that there is a quote that never again should people starve in a world of plenty. This particular statue was gifted by Ireland. And here you can see during the pandemic period, we initiated a program to distribute food materials to various places. As you can see, the people are collecting food in the queues. And also we have organized this Lab on Wheels program that was highly successful, keeping the COVID protocol in mind. And here you can see the students are carrying out work, keeping the COVID protocol. And here, how about the name of the series, Science Beyond Boundary, Invention, Discovery, Innovation, and Society? Well, if we have to serve the society, only invention and discovery are not enough. It has to go by this innovation. And that is how we have given the name. And how about Science Beyond Boundary? See, I am a professor of organic chemistry. And we started with this basic science in schools, and then science in 10+2, and then graduation chemistry, and then organic chemistry special in masters. And then even beyond, when one does research, then so many areas and it flourishes and flourishes. That means there is compartmentalization. So if one has to study in depth, definitely there should be compartmentalization. But unless there is like science beyond boundary, beyond compartmentalization, we will not be able to help the society. And that is reflected in the last year's Nobel Prize in chemistry, that for the development of a method for genome editing, genetics is just a tool for rewriting the code of life. Well, we also have speakers from various areas, like Professor Shanku Mollik, who will be talking on basic and applications, and Dr. N. Silva Kumar. He is the CEO of his company, DS Genosciences, and also Dr. Abhijit Mondal. He is a young scientist from the President's University. So we look forward to listening to them. So let us, make this platform for mutual exchange of ideas and sharing knowledge, coming up with newer ideas for the benefit of the society and mankind. And I thank you very much for your participation. Thank you very much. Mr. Prashant, please.
Yeah. Thank you, sir. Thanks a lot to our Professor Bach for your wonderful speech, which actually today establishes the main underlying theme of today's program. Now, it's the time to invite our Honorable Professor, Dr. Sudipta Chakraborty, Principal of Midnapore City College, to kindly deliver the welcome address. Sir, please. Okay, I think our sir is not present there due to some unavoidable reasons. Maybe our sir is actually not present. Okay, so now we will enter into our main scientific program. Okay, we are about to start our scientific program. In this webinar, we have 3-hour lectures. The first hour lecture will be delivered by Professor Shanku Mollik from North Dakota State University, Fargo, USA. And Professor Jayanta Kumar Roy from IIT Kharagpur will chair this session. So I am handing over to Professor Roy to chair this session.
Good morning, everybody. At the beginning, let me thank another former student of our institute, Professor Brojogopal Bach, for giving me the opportunity introduce one of our brilliant students today. So let me start. Professor Shokku Mullik did his schooling from Ramakrishna Mission system of schooling in West Bengal. First in Purulia Ramakrishna Mission, then Narendrapur Ramakrishna Mission, where from he did his higher secondary. And of course, clearing JEE with very high grade, he got admission to IIT Kharagpur in electronics. I will tell you just 1 minute, one very interesting thing. This was the trend that to be in top engineering branch was dream for many bright students, and he had a dream like that. But his love for chemistry was hidden. He has not mentioned this in CV. I will tell you. Kharagpur IIT, there is a program that in first year, if you do very good, some percentage of students can change branches. Trend was from science to engineering, not from engineering to science. And so far, my long career, I know only 2 persons. One is Shokku Mullik, went from electronics to chemistry in the second year, and another one, Kaushik Kaushik. So his love for chemistry started when he joined our department in second year, and we had long career, and even in 2019 we interacted. He invited me. I went to NDSU, and so we are in touch number of times. So I will tell you another very interesting thing. We had a 5-year integrated program that you know in chemistry, jointly based chemistry together. He did not complete that. After 4 years, that is equivalent to BS, he went to United States because of course clearing all the GRE, TOEFL, all those things, saved 1 year. Idea was that is equivalent to BS, no need of wasting another year in IIT Kharagpur. And he joined here from his academic career in chemistry, started strongly, that is at Case Western Reserve University, Cleveland. Almost same time, I was also planning to go to University of Chicago, so we interacted over there also. And he joined Professor Anthony Pearson, organic, organometallic, a little bit of inorganic chemistry, and did his PhD. And after that, he moved to the other side. That was the trend in USA. You do Atlantic side, PhD, postdoc in Pacific or vice versa. So he did his postdoctoral research in California Institute of Technology under a very famous scientist. You might be knowing his name. His name is Professor Francis H. Arnold. And that work was on bioorganic chemistry, from hardcore chemistry to bioorganic chemistry. So it was trained in different aspects of chemistry, biochemistry by this time. And you know, in 2018, the Chemistry Nobel Prize was won by his postdoc boss, that lady, Professor Frances H. Arnold. And after completing his PhD and postdoc, he got straight away a job at NDSU, or I should say, was probably North Dakota University. And all those stages, like assistant professor, associate professor, professor, he completed. And from 2009, he's a full-fledged professor of NDSU. And of course, in doing so, he has got many projects, bonds, collaboration, very big group. And I've seen 5 assistant professors being associated with him in 2019 itself. And his exposure is fantastic. Many students have got PhD, MS, postdoc working with him. As for statistics, I will tell you a little bit. That project from NIH, NSA, all this funding area, has publication more than 110, 8 index 38, citation 4,200, patents many, book chapters many. A long list is there. And I should mention that his interaction with me continued even if he joined NDSU. First thing, when he got the funding, he asked for any of my former students or students who is ready to join him. And it was Bidhan, who was very productive. And I visited his place after postdoc. He got a very good job. So his boss also very happy. said that Sonko also deserved thanks for that for him. And after Vidhan left, another student also joined him. That was Manos Haldar. And Manos Haldar also productive. I believe today he will tell something about Manos's polymer works. I am in touch with Sonko and Manos for a long time. And after Manos left, got another job, I said, Sonko, this time you hire my daughter who did PhD from France. So yes, now his group is very big. So she was accommodated over there and she was also productive and she is doing very good in USA. Regarding awards, he got many awards, as I should mention. The Teachers Award is fantastic. In 2001 also he got Teacher of the Year Award 2021, and he is fellow of many society. I just mentioned AIMB Career Award starting from 1998. That is National Science Foundation. I should not kill any more time because I said that his work is multidisciplinary, involves chemistry, biology, biochemistry, cancer, and today he is going to tell us a very interesting topic, that is polymer nanoparticle for drug delivery to hypoxic solid tumor. In these few words, I request Professor Sonko Molli, please start your talk.
Thank you, Jayantada. Let me see. So let me try this. I'm trying to share my screen and let me see on the PowerPoint. Okay, can you see my screen?
Yeah, it's coming.
It's coming. Okay, now, okay, what did I do here? Okay, I cannot see anything. Looks like I'm talking to a blank screen.
No. Now you have to go to PowerPoint. That one you have to share.
Okay.
That it is showing that you are presenting something, but you have to now share.
Okay, does that work now?
PPT.
No, we can see your slides.
Oh, you can. Perfect. Okay.
Only thing, probably you need to go for the full screen, and then it would be nice.
Oh, I thought I had it in full screen. Give me just 1 minute, please. Okay, let me start this all over again.
Maybe present mode, we have to do something with the.
No, I will try this. So let's try from here. Window. Yeah, I have to just select the right window. Presentation PowerPoint. Allow. Is it in full screen now?
Not yet, but it's okay. I don't know.
Sonko, carry on. This is okay.
Yeah, it's okay.
This is okay. What do you see? Okay, let me try one more thing. Just one more thing. See if this works.
Share presentation and.
Okay, what do you see now?
I think it's perfect.
It's perfect.
It's now perfect.
Okay.
Yes.
I got it. Thank you, Jayantada. Thank you for inviting me. And then I have to apologize to Professor Nag. This was just a really bad time for me with so many things on my plate. He must have thought that he's irresponsible, does not respond to my emails. I am sincerely sorry. Okay, so let me start by saying a little bit about what we are doing in polymer nanoparticles for drug delivery. And our interest is mainly hypoxic solid tumor. And before I do anything, I just want to start this with a dedication. That as Jayantada said, I'm a product of the chemistry department, IIT Kharagpur. And I want to dedicate this presentation to 3 of my professors who I still keep in touch, and I call them my mentors. First one is, of course, Jayantada. Second one is Jayasridhi. She helped me a lot in terms of career guidance and how to apply. And then Professor Pal, he also visited us from IIT Kharagpur. And for those of you who do not know, Tarashpada, outside his inorganic chemistry is also an excellent cook. So he actually taught me how to make a couple of sweet dishes. So with that, let me start by giving you some background of what I mean by hypoxia. What I mean is that here is a solid tumor growing, there is a blood vessel, but the blood vessel is only on one side of the tumor. If you go away from the blood vessel, the partial pressure of oxygen rapidly drops. And as the partial pressure of oxygen drops rapidly, these cancer cells genetically change themselves from a phenotype that can be killed by anti-cancer drugs to a phenotype which is resistant to anti-cancer drugs. And because these cells are surviving in very low oxygen content, I mean less than 2%, less than 0.2%, these are genetically different than the cancer cells on the surface. These cells are resistant to chemotherapy, and these cells are prone to metastasis. So the standard anti-cancer drugs will only kill the cells on the outside. These cells will survive, and that is a problem right now. So what we are going to do, or we are trying to do, is to prepare these polymer nanoparticles, which is tagged with a peptide. And I will show you the structures of these polymers. This peptide will take it deep inside a tumor, and it reaches a hypoxic microenvironment, latches onto the cancer cell surface, goes inside, sees a higher degree of hypoxia, higher degree of reducing agent, and then I will show you the organic chemistry. So that essentially explodes the polymer nanoparticles and releases the drug. So can we do it? How far can we take inside a solid tumor? And what are the consequences? So that's what I will show you in today's presentation. So there are all types of polymer nanoparticles. What we are going to use are called polymersomes. And I will give you a brief introduction. Again, you will see what I am doing in my research throughout is I'm taking my organic chemistry knowledge and applying it to a biochemical problem, which is relevant to a lot of patients. We are going to make polymers, which has two groups. One group is hydrophobic, another group is hydrophilic. We are going to link it with a small organic molecule, which is reduction sensitive. Now we have picked up this molecule such that the redox potential will match to about 0.1% oxygen or in hypoxic environment. If I take this amphiphilic polymer, put it in an aqueous buffer, then it gives a bilayer structure where the hydrophilic polymers are all laid out. They're interacting with water. Hydrophobic core or hydrophobic segments are inside, they're away from water. And if you imagine this bilayer to form a nice sphere, those are called polymersomes. So these are essentially bilayer vesicles. If I have a drug which is hydrophobic, that goes in the bilayer, which is right here. If I have a drug that is hydrophilic, then this hydrophilic drug will go inside, and that is the aqueous core. Second term that I'm going to use frequently are cancer stem cells. And these are, as I showed you in the illustration, these green cells in my artistic drawing, which are the core of a solid tumor. They do not, they can survive in low oxygen, and these are resistant to standard chemotherapy. Now, in order to get to it, there is a small molecule drug which is in phase 3 clinical trial for pancreatic cancer. It's called napabucacin, fairly hydrophobic. Here is the structure. However, this drug, there is no report which says that this drug can penetrate solid tumors and reach the hypoxic core. So our goal is, can we take this drug, take it to hypoxic core? Can I take a hydrophilic anti-cancer drug and take it inside a tumor and release them? And let's see what are the consequences. So common theme in my talk, polymersomes, I took you through. Hypoxic solid tumor, I talked to you about this. I'm going to briefly tell you about two of the cancers which we work with. One of them is pancreatic cancer, another one is triple negative breast cancer. So let's start briefly with the pancreatic cancer, and for the sake of time, I will go through this a little bit quickly. So the idea came from this paper which was published in JSCS 2010, that if you take azo dyes, so this paper essentially talks about small molecule azo dyes. you can do enzymatic 4-electron reduction in sequence. It goes through a hydrazine intermediate and breaks that nitrogen-nitrogen single bond to give you 2 primary amines. So I thought, can we do this on a polymer scale? Meaning, we will have a hydrophobic polymer, a hydrophilic polymer linked with a diazo linker, just like the diazo dyes. Can the enzyme still access these linkers, diazo group, even when it's embedded between 2 polymers? Successively put 4 electrons, first put 2 electrons to get to a hydrazine type of polymer, again 2 more electrons, and break it into 2 different primary amines. So that's how the whole project started. And this is where Jayanta, the student Manas, he came in, and I asked him that, Manas, because you have a training in organic synthesis, can we take these diazode linkers or these kind of dyes and link it to a polyethylene glycol on one side? So this is what he first did. He bought this particular molecule, which is diazo dicarboxylic acid. It looks sort of orangish red. Connected a polyethylene glycol on one side, took the other carboxylic acid and flipped it into a primary amine through a standard peptide coupling reaction, and then did a standard polylactic acid synthesis protocol with this lactone with a tin 2-base Lewis acid catalyst. And the first attempt he made, it had polyethylene glycol 2000 and the polylactic acid 6000. So we said, well, let's see what happens with this polymer. So that's how the project started. Next part of this is, how are you going to take these nanoparticles deep inside a tumor? So we looked into the literature and we found that a receptor called neuropilin 1, if we can find a peptide that binds to neuropilin 1, then that can go deep inside a tumor. There was just one report when we started this project. So we wanted to make sure that this receptor is expressed in pancreatic tumors. So we collaborate with a pancreatic cancer sort of clinic, hospital, resource center in Nebraska. And from them, we got 4 patient samples of primary pancreatic tumor and liver metastatic sites, 2 male, 2 female. All patients were primarily first went through a surgical resection followed by chemotherapy. And we are looking into how much of the neuropilin 1 receptor is expressed either in the pancreas, which is outside the tumor boundary. We don't see much. Okay, this is incidentally doing this Western blot here. Then primary pancreatic tumor, we see a huge expression of this receptor, and even in the liver metastasis. If you want to see it in a quantitative fashion, there is statistically significant expression of this receptor in the tumors and in the metastatic site. So here is a peptide that was reported. It's a cyclic peptide which binds to the neuropilin receptor, and then it takes it inside the tumor. So we thought we will synthesize this peptide and attach an alkyne to it, and that way you have a handle to link it to anything we want to. So we do this with a solid phase peptide synthesizer, which is microwave assisted. And you will see the effect of microwave here. Each of these coupling reactions, meaning any of these peptide coupling reaction, takes 135 seconds. So that's 2 minutes, 15 seconds. Each of the deprotection reaction takes 70 seconds. So this whole peptide, we can make it in 1.5 hours and then do a reductive cyclization. And then the product we find is nice and pure by mass spectroscopy. There is absolutely no need to do any more purification. Okay, so here is the next step. So we actually buy this particular polymer, which is the polyethylene glycol linked to an azide. And again, I told Manas, can we make the polylactic acid on the other side of this polyethylene glycol and see if we can get the molecular weight slightly more than the molecular weight you have? So he did some trial and error, get it to about 8,500 or 8,500. And then he essentially did a 3+2 cycloaddition reaction with copper 2 sulfate, again, a chelating agent so that it's not precipitating, reducing agent. And then we get this peptide in fairly clean fashion. Here is a spectroscopic characterization looking into circular dichroism. This is just the peptide in black. So that's the signature of the peptide. This is the polymer peptide conjugate. Here is the signature in red. Okay. So we started making polymersomes with the hypoxia responsive polymer. targeting peptide, and we put about 2% of this lysamine rhodamine lipid so that these polymers have little bit pink color, almost like this color, so that we can see, we can see the solution color. We encapsulated the standard drug to treat pancreatic cancer, which is gemcitabine. And then we also encapsulated the napabucacin, that's the drug to kill the cancer stem cells, in 2 different formulations, and then check their release in normoxia, which is 20% oxygen, hypoxia, which is 1% oxygen. In either case, you will see the drugs are reasonably stable in the polymersomes in normoxia. With 1% oxygen, it rapidly releases the drug from these polymers. So then we checked that if we take this drug and treat it with pancreatic cancer cells, does it really kill the cells? Again, I have a lot of biography, a lot of data, a lot of statistics. All I would draw your attention to is if we have the hypoxia responsive polymer with the gemcitabine, hypoxia responsive polymersomes with the napabucacin, that's the brown bar. That's where we see the viability of these cells decrease to about 25, 26% here. And these are actually, when we are not a monolayer culture of the cells, so the cells are growing like a spheroid. So this is about 250, 300 micron in diameter. And these are the cells. When we treat it with these two together in hypoxia for 72 hours, essentially the entire solid structure of this cultured tumor disrupts. You see these smaller fragments. Again, anything red means dead cells, anything green means live cells still. So after 72 hours, we see a lot of dead cells and total disruption of that solid tumor mass. We also wanted to check whether they are penetrating these solid tumors. We actually purchased some cancer stem cells, and these are derived from patients undergoing surgery. Grew spheroids out of these stem cells, treated them with the polymerism. So here is the boundary of these spheroids. Now let's look into this figure first. If we don't have the peptide, most of the polymerosomes are on the surface of these cultured tumoroids. If we have the peptide, again, we see some of them in the surface, but we see a lot of them going inside these spheroids or microtumors. Okay. Then we also wanted to check, so if we take these spheroids and then look for other stemness markers to make sure that these spheroids do have cancer stem cells. And in this case, we are looking into a marker which is called CD44. And anything in violet, I don't know how good the colors will go, so I'm going to put a circle here. So this is the one where we see the stemness is decreasing. So let's start here. If we grow the spheroids in hypoxia, you see a lot of these pink colors. So that means we are getting a lot of CD44. And that shows that we are getting a lot of cancer stem cells in hypoxia. We treat it with the hypoxia responsive polymerosomes just with gemcitabine and hypoxia responsive polymerosomes with the napabucacin. And you will see the pink color is decreasing. And in this case, there is hardly any pink color left, telling us that these combinations will reaching the stem cells and killing them. Okay, I'm going to skip over this for the sake of time and show you some data on some mouse pancreatic tumor. So here is some data with mouse pancreatic tumor. So we grew this pancreatic tumor in the pancreas of the mice. And here is one such tumor, about a centimeter, centimeter and a half in diameter. To show you, if I take this tumor and slice it with a scalpel, it's a fairly solid block of tumor. And there is hardly any blood vessels, so there is nothing red inside. It's just a solid fibrous mass. Then we treat these mice with our polymerosomes, and in this case, you can hardly see the blue. So essentially this blue, which is from these polymerosomes, are getting all the way there, sort of blue-green if you see this here. Blue-green means it's the polymerosomes. So if we treat these mice with an intravenous formulation of these polymerosomes, they essentially go inside this solid block of tumor in the mice pancreas. And if you take any measurements, it says 3,220 micron or 3.62 millimeter in here. This is about 10, 12 millimeters. So we can reach about 4 millimeters from the surface. So it is penetrating substantially in the tissue. And without getting into all the details here, if we inject these mice again intravenously through these formulations, only thing I'm going to draw your attention to these pink circles. So we can control the growth of the tumor even after the treatment ends. And at this point, we sacrifice the mice, so we don't know what happens after this. But up to 6 weeks, we can control the growth of the tumor only with the two polymersomes, hypoxia and responsive together. All the other tumors essentially grew right after the treatment ended. Okay, so again, briefly for the remainder of the 10, 12 minutes, I wanted to show you our the triple negative breast cancer. Again, here's the polymer that Manas made, and this time I wanted to see what happens if I take the carboxylic acid from the para position. Para means para with respect to the diazo link, and move it to a meta position. Does it have any effect in forming the polymersomes in terms of their stability, in terms of their reduction potential, and do they do the job? Again, same chemistry Manas went through. So here is the polyethylene glycol amine that we can buy, link it to one side carboxylic acid, take the other carboxylic acid, and then change the acid to a primary alcohol through a standard peptide coupling, and then grow the polylactic acid chain. Exactly same chemistry. In this case, it's 8,500 polylactic acid, close to 8,000. So I said, that's good enough, Manas. You just scale it up, purify the polymer, make sure it's clean, make sure by NMR, by gel permission chromatography, by mass spectroscopy. And once it's clean, then we are good to go. So by gel permeation chromatography, it looks fairly clean. Polydispersity index 1.17, which is fairly good. And that is, again, the hypoxia responsive linker. Okay. Quickly show you in this case, because it's triple negative breast cancer, we are going to encapsulate doxorubicin in the polymersome. That's the hydrophilic drug, standard chemotherapy to treat triple negative breast cancer. We can release the drug under 1% oxygen, but normoxia 12 hours, it does release about 30% of the drug, but not 90%. Okay. Here is some structural studies. So right after 12 hours, if we take this polymersome and look under atomic force microscope, we still see these nice spherical shapes. If we take these polymersomes in hypoxia after 12 hours, do the same atomic force microscopy, there is essentially no sphere left. So again, as we reduce these diazo link to two primary amines, that leads to a structural collapse of these spheres, and we see essentially a complete loss of structure. Okay, okay, I'm going to skip over this whole thing and show you some other interesting examples here. Okay, so here is an interesting example. If we culture triple negative breast cancer cells as monolayer culture, and then see if these polymersomes are any better in killing the cells with doxorubicin, we see that at 4 micromolar concentration of doxorubicin, that's what 4 stands for, with the IRGD peptide, that's the penetrating peptide, we see the highest difference between cell killing ability between normoxia and hypoxia. We have done quite a bit of biochemical studies in what's the mechanism, and because we are delivering doxorubicin deep inside, we are not changing the cell killing mechanism of doxorubicin. So the doxorubicin cell killing mechanism remains intact for all of these formulations. Okay. We can do exactly the same study with microtumors grown. So these are about 300 micron diameter from the triple negative breast cancer cells. Again, the penetrating peptide, 4 micromolar doxorubicin, highest difference in cell killing ability for the hypoxia and normoxia. And here is a microscopic picture. So once we treat it with these polymersomes under hypoxia, we see these dense microtumors begin to loosen up and fall apart. Okay, I'm going to skip over this for the sake of time because I wanted to show you something interesting. So we can do this experiment in mice, and I will draw your attention to these sort of like purple diamonds here. And that is the peptide-targeted hypoxia-responsive polymersomes with doxorubicin, and that controls the growth of these fairly aggressive triple-negative breast tumors in mice. Okay. We also, again, we are killing the mice anyway. So we excise out the tumor, and this is how the tumors look. Again, the targeted polymersome. So that's these data points here. The tumor volume is lowest. And since we take the tumors out anyway, we measure them or weigh them, and the weight of the tumor is also least when we have in this treatment point, meaning the targeted polymersome giving doxorubicin inside the tumors. Okay. So where are we now? So I just wanted to spend a few minutes on this, which is the translational aspect, and then stop there for any question. And that is now we are actually teaming up with an oncologic surgeon, Dr. Daniel Tuvin. He does surgery on either triple-negative breast cancer patients or pancreatic cancer patients. So we have set this up such that if a patient has a surgery all planned, then on the day of the surgery, about 2 hours before the surgery, Dr. Tuvin's nurse will call us. And as Dr. Tuvin's nurse is preparing the patient for surgery, we prepare 2 or 3 mice for surgery at our side. Of course, patient has to agree that a part of the tumor which is taken out after pathology can be donated to our team. Once we get a call from the surgical nurse, from Dr. Tuvin, that yes, the patient agrees and Dr. Tuvin will take, let's say, about 3 hours for the surgery. So then we have everything ready on our side, and we are there to get the tissue from his surgical nurse, take it in our animal surgical facility, and put it under the skin, depending on how big the tissues are. Usually they're about 0.5 millimeter by 0.5 millimeter, 0.5 centimeter by 0.5 centimeter by 0.5 centimeter. We slice it into 2, put it on this side one, put it on the other side, and let the mouse recover. So that is the first phase, which we call F1. Let the tumor grow, and these are immunodeficient mice. So these mice will grow the human tumor and keep the genetic signature of the patient intact, meaning the tumor is not genetically changed. Once the tumors grow to about 1 centimeter, and that takes anywhere between 3 to 5 months, depending on the stage of the tumor, and of course, each patient is different. Then we euthanize the mice, take the tumor out, slice it into 4, and again, put it into 4 different mice. And we essentially propagate and expand this patient tumor, keeping the genetic heterogeneity intact. So right now we have, we have 6 so far, but these 3 are growing best, these 2. So we have a tumor from a 34-year-old female who was pre-treated with these drugs, and a 69-year-old female who was not treated with any breast cancer drug yet. I'm going to show you the data from this particular patient who was 69-year-old at the time of surgery. She did not receive any chemotherapy drug. And I wanted to see that does she have any stem cells in these tumors once we grow them in mice. So again, we are looking for the CD44, that's the marker. And if you look into the core of these tumors, again, grown in mice, we see a lot of this orange color, meaning the tumor that she had a lot of stem cells, and she had a very metastatic version of this tumor. Again, the stroma also has a lot of CD44 positive, again, showing that this tumor is very, very aggressive. Okay, second thing we did was, because this tumor had CD44, which is a stem cell marker, so we wanted to see if it has the neuropilin-1 expression also. And we did see, again, for this particular patient, the core had a lot of neuropilin expression, and the stroma has a lot of neuropilin. I did not take you to the other patient. Turns out the other patient had high CD44 expression, which means the tumor is pretty metastatic and aggressive, but did not have a good level of neuropilin-1. So right now we are checking if we can fine-tune these formulations to meet these patients' individual needs. So that's where we are. That's where I'm going to stop and then make a few acknowledgements. It's primarily supported by a grant from NIH and another grant from EPSCoR. There is another NIH grant which essentially encourages the collaboration between organic chemists like me and then clinical oncologist and surgeon like Daniel Tuvin. So that's what sets up and keeps this collaboration going. I work with Satish in our group, not our group, our department. He's my colleague and he gives me all the biochemical insight. Tony Hollingsworth has a huge collection of human pancreatic cancer samples, and we get some from him from time to time. The polymers were started by Manas, and since Manas left, then Lee started making all these polymers. And then Jagadish and Nimesha, they helped us with all the surgery to take the human tumor, put it in mice, make sure they are growing. And then Jody used to be the animal core facility director. And my students who do it most of the work, Babak, he graduated. He is now a postdoc at University of Oregon. Matt graduated. He is now a postdoc at a research center in Sioux Falls. And right now, Jacob and Connor, they are now working with these patients. So with that, I'm going to stop. And I would be happy to take any question. I will also say that it's 11:30 p.m. here. So after the question answer, I will probably go and take some rest. So any question?
Okay, thank you, Shankhu, for your very nice talk.
Why I had to hurry up a little bit, but I wanted to give.
No, you had time. You can continue. You have completed your time.
So the flavor I wanted to give is that with good training in organic chemistry, and I owe it to you, good training in organic chemistry, you can apply that knowledge to solve some meaningful problems, and especially then start working with real patient samples and start collaborating with surgeons to get a really translational aspect. But again, crux of it is for all the students and everyone and the researchers. The crux of it is I'm an organic chemist. The crux of it are polymers, which are essentially doing a 4-electron reduction, which essentially takes the diazo group and makes it into 2 primary ends. That's the crux of the chemistry.
Okay, Shankhu, I believe if you can take 1 or 2 quick queries, that will be very much helpful. So now it is open.
Can I?
Any question? The Projo has
Go ahead, please. Go ahead, please.
Professor Ray, can I ask one question?
Yeah, sure.
Oh, yeah, I think Projo is having a question. Projo, please go ahead.
Go ahead. Go ahead, please. Go ahead. Yes. Yes, go ahead, please.
Yeah.
Okay, I'm sorry. I think he's taking some time.
Yeah, now he's okay.
Yeah,
No, you have some problems with your.
Yeah, there is one new problem.
You are using your polymer, right? To
Yes, I
carry
mean
the
polymer,
drug.
yes.
To carry the drug.
I got caught up. Can you say that?
The drug, right? To carry the drug to the site.
Yes, we are using the polymers to give the drug to the site. Yes, exactly.
And what happens after the effect?
Meaning what happens to the
remove the
polymer and
polymer.
what happens to the drug, or what happens to, I will say both.
To the polymer.
Polymer. So the polymer essentially gets degraded, I mean, reduced into a polyethylene glycolamine. And polyethylene glycol, what FDA, it's used all over the world as a laxative before colonoscopy, or if someone has severe constipation, that's the drug. So it essentially gets excreted. It is not metabolized and it's non-toxic, so it gets excreted. And the other part of the polymer is polylactic acid. That's a biodegradable non-toxic polymer. So those are the self-dissolving stitches the surgeons put in during a surgery. So it will hydrolyze over a period of 1 to 2 weeks, give you lactic acid, and that's purely non-toxic. We generate a lot of lactic acid in the body.
Okay. The thing is, we have got some natural products which are also forming vesicular self-assembly. And like say this oleanolic acid and then organolic acid, and they are partly water soluble, and DMS water they are soluble, and they are forming vesicular structures. And also we have been able to encapsulate some drug molecules like doxorubicin, and I hope they themselves have some anti-cancer activity, but we are waiting for some collaborative work so that we can try those trials on animals, because as such in ecosystem they are forming vesicles, and they can also even entrap doxorubicin and other fluorophores inside. And
does it release the doxorubicin? And
Yes,
what
yes,
would be
yes,
the release
yes,
like?
yes. They encapsulate and then they release also in page buffer. Yeah. And maybe I'll send a
Okay,
couple
so
of papers to you. Okay, then.
sure, maybe what I was going to do, I mean, I was going to say that maybe you have my email address.
Yes, yes, yes.
Yeah, let's set up a Zoom chat. I'm much more familiar with Zoom. Give
Sure.
me a few days and times. And next week is very busy for me. I have the classes are starting. So not next week, but the following week. Give me some dates and times when you are available, and I would be happy to do it over the weekend too. I will set
Sure.
up a Zoom link and let's have some 1:1 discussion.
Yeah, sure, sure, sure. Okay. All right. Thank you. Very nice work.
Oh.
Excellent.
Oh, well.
I liked it.
Professor,
Any
can
other short question? Any other
I have a short question? I
Yeah,
have a
please.
short
Sure, go ahead. Yeah.
Yeah, Professor Malik, it's fantastic talk as the organic chemist turned into cancer biologist. Is something which is a big
I'm
transfer.
not a biologist. I make this distinction, right? You can't change my
You are
training.
a chemical biologist. I think you are a true chemical biologist, I would say, more than a cancer cell biologist. Thank you very much for the talk.
Oh,
And
yeah,
I'm just wondering, actually, CD44 is a glycoprotein. Actually, it's a, there's a cell membrane surface to surface, this aggregation and all those things happening. So did you really, or are you looking into the matrix metalloproteinases? Because that is also having a, effect on this CD44, because CD44 happens to be a aluminic acid, receptor, actually. Thank you.
Yes, yeah, we are looking into MMP2, 9, 13, as
Gelatinase.
well as
Gelatinase. Okay.
Adam 10. Because Adam 10, these are also these extracellular matrix metalloproteinases, which are responsible for cancer metastasis. So yeah, we are looking into those also in these patient samples, both the total amount of these enzymes through ELISA and the amount of active enzymes through an activity-based assay. Yes, I don't have that data yet, but my student Connor is doing exactly that.
Thank you so much, Professor. Thank you.
Welcome.
Thank you, Shonku. Very nice presentation. I will not repeat those things. It involves chemistry, biology, cancer, prevention of cancer, protection, all those things. So I'm really thankful to you for accepting our invitation. And also another request is still pending. I hope you will complete it within a dot two. Okay. Thank you very much for your nice presentation.
You are welcome. You are welcome. Thank you for inviting.
Stage is for you.
Oh, thank you.
Yeah.
Thank you.
So, based upon your scientific achievements and also for the popularization of science, like the society CRS offers you this gold medal, and also
thank you. I'm honored. I'm honored to receive the medal.
Thank you very much. Thank you. And this is the certificate. Okay, from the
Sure.
president.
Thank you so much. I mean, you can please send it to me as an email because you have the certificate picture. And then send me a few dates and times when you are available, not next week, as I said, but in fact, if you want to do it, the first weekend in September, that's a holiday here. We can do it during that weekend. So let me know if you have some time. This will be, I think, September 4, 5, and 6.
Okay, sure. Very nice. Excellent.
Okay.
Congratulations.
You are welcome. You are welcome. And since you have a few people from Vishwabharati, I know a chemistry faculty there quite well, and that is Gautam.
Okay, okay.
So
And today we have Prithvideepa Sahu. She is also doing excellent work, and today she performed that vocal song, whatever she performed today. Prithvideepa Sahu. She is also
that's very much. She sings very well. I was going to ask which raga is that. Not that I follow all the ragas, but
Thanks a lot. Well done.
You are welcome. Again, as I said, it's 11:40 here, so I'm going to log off and take some rest.
Yeah, pleasant for us too. Thank you. Excellent.
Thank you. Thank you again for inviting me, and I'm honored. Please send me the certificate by email attachment. And send me a few September 4, 5, 6 if you have some time.
Sure, definitely. Yeah.
Okay. Good morning, everyone.
Good morning. Good morning. Thank you.
Mr. Pradhan, please. Mr. Prashant Pradhan.
Yes, sir. So thank you, Professor Malik, for your insightful presentation. Okay, so now we are in the next our lecture, which will be given by Dr. Selva Kumar Natsan, DSK InnoScience Private Limited. and Professor P. Jayasankar, Fulbright visiting scientist from USA, will chair this session. I would like to request Professor P. Jayasankar to take over from here.
Thank you, Dr. Pradhan, and good morning, everybody. And at the outset, I would like to thank Professor Rajagopal Bagh, the convener of this symposium and the president of CRS, for organizing this kind of award lectures, our series of award lectures on behalf of the Sansthan. And it's my great pleasure to be always associated with RASAYAN. And today, we are going to see another transformer, actually. Just now we saw how Professor Malik, being an organic chemist, he has transformed into a cancer biologist and probably as a chemical biologist in general terms. Now, this lecture is going to talk about how an organic chemist turned a successful entrepreneur. What a beautiful combination of the selection of the speaker, Brajo, and I think hats up to you. And it's my pleasure to introduce Dr. Selvakumar Nadesan. Dr. Selvakumar is the CEO of DSK InnoScience Private Limited. He's in Osur. And he is a trained organic chemist. And he is the promoter and managing director of the company, DSK InnoScience. And the InnoScience has got a fantastic facility for R&D with a modern chemistry lab and analytical lab. It is also having kilo facility to undertake scale-up projects up to 500 kg. This is the fantastic facility which he has created, actually. Before starting his own DSK InnoScience, Dr. Selvakumar was associated with the Anthem Biosciences as a vice president in the drug discovery program. And in fact, he was heading more than 200 scientists, actually, So that was a big success story of Dr. Selvakumar. And even before joining Anthem Bioscience, Dr. Selvakumar was with Dr. Reddy's, and he was the head of several scientific and leadership positions in the drug discovery program. And he played a management role for the discovery of the talents, and recruiting the people. And also he played a discovery of the teams of about 6 molecules in the development and coordinated with the biology and the medicinal chemistry people. So Dr. Selvakumar, before, during his PhD, and he did his PhD from Indian Institute of Science, and he moved to USA and Canada for his postdoctoral studies. Dr. Selvakumar did his schooling and graduation from Tamil Nadu, from Trichy. That's a beautiful place, actually, St. Joseph College. I did his MSc from the St. Joseph College, Trichurapalli, and PhD from Baridasan University, the MPhil from Baridasan University, and he did his PhD from Indian Institute of Science, Bangalore. So he was holding a lot of responsible positions in the both industry, and he was a true academician, I would say. Like us, he has published many fantastic papers and very important papers, actually, publications in the medicinal chemistry field. Not only that, pure organic chemistry field. Actually, I think he has completed total synthesis of 50 steps, involved with 50 steps. That's a remarkable achievement. I think probably Nicholas' lab is very famous for, going for total synthesis of natural product chemistry. I think that's a fantastic one. I think, he has done the remarkable job. And he has published many synthetic organic chemistry related papers in the chemical communication, organic letters, Journal of Organic Chemistry, Journal of Medicinal Chemistry, Bioorganic Medicinal Chemistry letters, etc., to name a few. And also, he has filed many patents to his credit, actually. That's what he's having experience both in academic as academician as well as entrepreneur. So now we are going to see how Dr. Selvakumar transformed himself probably to become a successful entrepreneur, being a organic chemist. With this small introduction, let me invite Dr. Selvakumar Nadesan to present his presentation today, the award lecture number 2. Thank you. Thank you very much.
Yeah, thank you, Dr. Prasuram. Thank you for a wonderful introduction. First of all, I would like to know whether everybody is hearing my voice. Is it okay?
Yes, fine.
No problem.
Okay, because I am traveling now. I'm in a remote location, but thanks to the internet, and if everything goes well, I should be heard properly. So I think to begin my speech, I would like to also thank Dr. Brajagopal Bag, who was my colleague at Indian Institute of Science, very humble and very hardworking colleague actually, but he was in my neighbor lab actually. So I remember those memories, and he called me and he asked me to give this lecture. And see, what I did today, before giving my speech, the presentation I made is first time I made this presentation. In fact, every letter in this presentation was prepared by me in the last 1 week or so, and the slides are brand new, and I am giving only here for the first time. So if there are some mistakes, you please pardon, but I will try my best to tell what Dr. Prasuram was mentioning actually. So I'm going to upload my slides. Is it uploaded? I don't think so.
Yeah, it's visible now. It's visible, Dr. Selvakumar.
Oh, very good, very good. So I didn't know. Okay. So if the slides are seen for you, then can I continue my discussion?
Can you please hide that stop sharing button down? Hide.
I think it goes off automatically. I think it goes off. Yeah, thank you. Now it's gone.
Oh, okay, great. Okay, so yeah, basically what I'm trying to say in this particular presentation for the next 20-25 minutes or so, I felt that in a country like India, we are given great education, but how to transform into an entrepreneurial path is something that we should inculcate in the curriculum of the education in India. That's what I felt after so many years of experience in both academics and industry and then entrepreneurship. So I'm trying for the first time, as I told you. And are you seeing my second slide?
Yes.
Yeah, I'm just confirming one time. So here in the second slide, I am trying to give the difference between Indian and Western education. Basically, these are all my own thoughts, and not to offend anybody. But basically, if you see the Indian education, at least in the southern part of India where I am coming from, we are mostly taught, the education by reading books. We are asked to read, and then we are asked to score in the exam, and we are given celebrity status when we score the state first, and we are revolving our entire education around reading books. And this, according to me, this teaches us the art of playing safe. That means you are aware of only reading books, and subsequent to your education, you are interested in taking your career by playing safe. And our Indian education is teaching, the celebration of success, but the most important thing is it is not teaching, it is giving you, it is installing the fear of failure. This is the most important thing. So if you want to take an entrepreneurial path, you should not have the fear of failure. But our education did not teach that. We are always, we are always celebrated if you score higher marks. On the other hand, if you see the Western education, because I lived in the West for about 4 years, and I'm traveling to the West very, 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 very frequently after seeing them very closely, I can say their education, there is no social stigma. The society does not expect anyone to become so and so. For example, if you see the geography where I am coming from, there is a huge craze for becoming a doctor or becoming an engineer in the past, and something of that kind. These are all social stigma, but this is not there in the Western education, and there is no boundary in the thinking process. The students, many of my friends who are NRIs, their children grow in the West, and when I see them very closely, I felt they don't have any boundary in the thinking process. They just don't have any boundary. And most importantly, the Western education is inculcating the spirit of risk-taking ability. They don't think that we cannot do certain things. And most importantly, they take success and failure as the one. This is the only beauty of the Western education, is making, it is making the Westerners to aspire for bigger things, and it is making Indians to play a safe game. So having said that, in the next slide, what I'm trying to say, this is my personal experience in the last 4 or 5 months. Coming from Indian Institute of Science, the Department of Organic Chemistry, where I did my PhD, the alumni of the department approached me that we can all, form a WhatsApp group. Nowadays, WhatsApp is the big buzz, and I immediately obliged. I could see in a very short time, more than 300 people, who are alumni of the department have joined. And on one fine day, we were all asked to introduce. So each one of us introduced. I was going through, profile of every organic chemist graduated from the celebrated institute, IISc Bangalore, and I found, to my surprise, the faculty positions, they are holding faculty positions across the country. Please don't take me don't get offended. But what I'm trying to say, from when I saw the profile of all of them, they were holding faculty positions across the country in India, whether you call Trivandrum or you go to Jorhat or Bhopal or anywhere, Jammu, and some of them, they were successfully employed in overseas companies. If you see, particularly the United States, they are all employed in very good positions in the United States. But I could say no one is successful entrepreneurs. That made me to get surprised. Okay, see, very, very celebrated institute. There is no institute better than Indian Institute of Science in the country. But, the people are mostly opting the safer jobs. But in the West, if you see, when I went to the West first time, it was in 1995, I went and landed in Canada, and I had a health problem. I went to the university. I see the doctor has given me appointment at 2:30 p.m., but they say he is right now in the laboratory doing research. See, that is something in India I don't see. See, a doctor who is going to do treatment for me is doing research in the lab before coming to his appointment in the West. But in our country, doctors are very busy in writing all the time prescriptions and doing operations. So this is what, this is what opened up my eyes. And you can see in the Western education, the students and the professors start companies. The students are doing their graduate studies or postdoc, and the professors are full-time professors, but all the time they think about, commercializing or taking in the entrepreneurial side. And I can tell you, many of such professors and students I know who are right now entrepreneurs in the overseas companies, they are with whom we are working from our company. That is why I'm able to say very clearly that there is a phenomenal difference between the Indian and the Western education. So what is the suggestion from me? The suggestion is, the educational system in India should should move away from the book-based reading, and half the time should be dedicated for non-book-based, something like workshops, the one which like what we are doing now, our seminars, are new project proposals. I think a student, while doing a graduate studies, can come up with the new project proposals, and that could be all deliberated in the department. and that could become a starting point in the future. And similarly, the entrepreneurship brainstorming. I just mentioned basically entrepreneurship, but basically brainstorming sessions. So the students, and after getting guidance from the faculties, they should sit in several brainstorming sessions. They should talk about, they should talk about the society matters. They should talk about worldly matters. They should talk about the subject. There should be several brainstorming sessions rather than you are glued to only research all the time. And they should have a lot of educational tools. And only the remaining 50% of the time, according to me, the students should be spending time for learning the subjects. And this is what pretty much I have seen in the Western country. So what is the solution I am giving? Basically is a startup ecosystem. So here I want to tell you very, very important. The first point, the startup incubators everywhere like Israel. So this point I want to talk for 2 minutes. It is so important for us. See, Israel, I want to tell you that in my business experience, I had an opportunity to travel to Israel very frequently. And each time I go, I spend about 2 weeks. And I must have gone several times. And after seeing the country very closely, I want to share with you. See, the country Israel is a very small country. And I think the size of the Israel must be equal to the state of Goa in India. It's a very small, tiny country. And most importantly, the citizens of Israel, they don't have a peaceful time. They are all the time, living in scare. And, the bomb can come and kill you at any time. So that is the kind of unfortunate situation the Israelis are living due to the Palestine and Israel struggle. And since I have visited several times, even I have gone through such things. But what they did, they realized that to live in a world like this, we have to be entrepreneurs. We have to create. We have to create. We have to form intellectual property. We have to protect them, and we have to be successful. So I could see the young students, young kids of Israeli, educational system. Even in coffee board, in a common place, they sit and talk about how to start companies, how to aim big, something like that. It was really surprising me, and that is the reason why today Israel has been a phenomenal success in the worldwide. And I can see the West, both Europe and the United States, they all the time travel to Israel to find out what technology they have for their problems. So right now, Israel has been recognized as a technology hub where people innovate, people, become entrepreneurs, very young kids, selling their company for $200 million. So that is, after seeing that, I felt that in India, we have to inculcate a startup ecosystem, and we have to encourage risk takers. And you see the fourth point I'm telling, imparting visibility for the achievers. See, what I'm saying is, you see, in a country like India, it's a large country. See, we celebrate, the people who succeed. For example, in the recent context, I can say that Neeraj Chopra, he got the first gold medal in athletics, and he is being celebrated in the country so much. I feel, an achievement of people like us, when they become entrepreneurs, their effort is no less compared to the Neeraj Chopra. So in the country, there should be an ecosystem where even the common people celebrate, or they treat, the successful entrepreneurs as celebrities. So such kind of a culture is required in the country. It's a phenomenal shift in the way we live in the country. I am proposing actually, and by having said that, in this slide, what I want to tell you, what is the joy of an entrepreneur? As Dr. Prasanam said very nicely, that I had been a strong scientist, I had been a strong organic chemist, published several papers, and subsequently I did medicinal chemistry, drug discovery, lot of research. But at the end, when I became an entrepreneur, the joy has no bounds. So that I want to tell you in this particular slide. See, by becoming an entrepreneur, what am I doing? I am not just seeking jobs. I'm not going and attending interviews. I'm not asking job. I'm not asking other company to give me a job. And most importantly, I have no craze for government jobs. I see in the geography where I'm coming from, the South India, a lot of young kids spending 5 to 6 years, attending coaching centers. What for? To get jobs, government jobs. The group 1, group 2, group 3 kind of administrative jobs, or you name anything. Even for passing your CSIR examination, there is a coaching center. So lot of craze for basically government jobs. I don't have that. And with a little bit of hard work, innovation, and differentiation, at the end, what I feel is the satisfaction of giving jobs. I think, right now, our company DSK is employing about 100 people. And when I see 100 people getting employed by one small organic chemist, the kind of satisfaction I have today, I have never experienced in the past. Tomorrow, we may be a 1,000 crore company, but that would not make me happy. But if I see 500 people getting employed in the process, that makes me so happy. There is no happiness, in my opinion, in my career of, now I am 55 years old. In my career, I have not experienced this happiness in any other thing, although each and every aspect of my career journey was very, very enjoyable. But this is a very different experience. With that, I want to tell you, the emergence of China around us, all of us, please watch, we quietly missed seeing this country emerging just behind us, just in the backyard of India, actually. It has become superpower, and they aimed big, not long ago, something like 20, 30 years back, with their all-round performance and relentless hard work. Today, they are world leaders. We are all, glued to the West, but we forgot that our good neighbor has become the superpower today, and he is dictating the terms. But on the other hand, in India, we don't have unity. There is so much of difference. By looking at each person, we are looking at who are you. And in the process, after independence, the interest of country was compromised because we have so much of differences in opinion amongst ourselves. We did not work like a country. We did not think the way China has thought. With that, for a vibrant India, what is the suggestion I am giving? For the vibrant India, the suggestion is self-reliance. We have to simply sit down and then think where we are not self-reliant, where we are importing maximum, where we are exporting less. So there we have to concentrate. We have to become self-reliant. I think if another country can manufacture an item, a product, which India is importing relentlessly, I think we should believe India also can manufacture the same product and export it in 20 years. So this is what is my feeling actually. And in the process, there are two things I want to tell you below. That is, one, cities are no more ultimate. This point I want to communicate with the success of DSK. I want to tell you that cities are really crowded. There are too many people, and the infrastructure of the Indian cities is not to cater such a large population. And gone are the days that only cities give all the infrastructure required to do something big. So that is where I thought, we will travel in a different route, and then we will establish a company in a village. We have done that. When we have done that, people thought that, I think Selva is all the time thinking crazy. And how is it going to be possible? But today I am telling you, we are in a village. We are working for the grassroots development. We are employing 100 people, in which 30 of them are from neighboring villages. All of them are getting decent money, and they are living decent life. So I think we have to move out of cities, go towards villages in order to make the country prosperous and self-reliant. This is my personal thought. And I think in the interest of time, I don't know whether we have time or not, but this is about, I have ideas wherever I think, and also I should tell you, I'm basically from a farming background. I'm a farmer, meaning my fathers were farmers, and I was thinking what to do for our farming community in the country. India is largely a farming country, so the farmers also should be successful. So for that, I am proposing this idea. I will make it fast. What I'm thinking is a group of youngsters, educated people in each village, they should form a team and they should pool the available land from the farmers. This is what my dream. We have to pool all the lands. Whoever farmers are not interested in farming seriously, and then make a large chunk of land, make a task force and do group farming, do a team farming, and bring in technology, select only 2 or 3 crops, to mitigate the risk. You select 2 or 3 and go for farming, make big money and sell very big, maybe export possible, and then share all the income amongst the contributors of the land. Like that, I think it is very much possible. I don't know whether in the country it is happening, but I feel, if from a farming background, if I do, I'll be making it successful. This should be the way in the future our country should be transforming. With that, this is the most important slide. This may be my last slide of my presentation before I give you a few slides with some pictures about our company. So what are the business opportunities in chemistry? I want to tell you, chemistry is one of the fantastic, subjects, sciences, where a lot of opportunities exist in doing business. I think, say, for example, if my friend is studying MA English literature, I cannot expect him to be an entrepreneur as much as I can expect my chemist, my fellow chemists to become entrepreneurs, to pursue the path of, business. So some of the thoughts I have put in here, for all of them, I have ideas. I think, in the future I should do that. But these are all my thoughts, and definitely there are opportunities. For example, one of my friends, he was a chemist, and from marine source, he found one compound existing more, and then he found a method to extract it, and then now he is a millionaire now. It's a real example. And there is abundant need for raw materials in the country. How I am telling, because running the company, I see all Indian companies are largely dependent on China to import raw material. I think this is really not acceptable. And as a small company, DSK, we have put our step forward in stopping whatever possible thing we could do in purchasing from China. We are stopping it now. We are making our own raw material. I think there is a huge need. If you go and see the importing mechanism in the country, a lot of raw materials are imported from China. That we should stop it. And there is a large potential existing in botanical sources. With the rich diversity coming from a tropical country, the botanical source of the country is very rich, and a chemist can explore an option of becoming entrepreneur in this path. And a lot of opportunity exists in material science also. There's so much in material science. And maybe we are, by virtue of our business model in our company, we are working with all these kind of collaborators overseas. We predominantly work for overseas companies, and we have collaborators in each of this segment, actually, that I can say proudly. And also the last one I want to say, the vast pharmaceutical phase. The pharma, today, I think we all hailing from India, Hyderabad, Andhra Pradesh has become the pharmaceutical capital for the country. And not only for the country, it has found a solid place in the world map of the pharmaceutical development. Today, you name any drug, it is getting exported from Hyderabad to any country in the world for in the API segment, actually. So the pharmaceutical space, it has not spread outside Andhra Pradesh. It should go. That's what I thought. I spent 10 years in Hyderabad. After that, I thought, okay, let's take the first step forward, move towards where we did education. So I was in Bangalore, getting my graduate study, PhD. Then I moved this side to start something else, so something like this. So I feel that the business opportunities exist in all these areas, and there's so much we can talk. I myself have ideas to do something of this kind in the future. I want to do a lot, with my another 10 years I can work. I think I will be definitely doing in these segments, at least another 1 or 2 ventures I will do it. So there's a lot of opportunity exist for chemists, to become entrepreneurs, and please believe that we can transform. We can transform the way our country is going today. And as I said, that is my end of the previous, the slide. Sorry, the previous slide is the end of my lecture, but 2-3 slides I will take it to probably show about our company. The picture what you see is actually the actual picture of our company DSK, and basically we say we help to make your molecules. So what we do, we make any molecule which is in the chemistry area, and we are very successful in making molecules in our company. And the guiding philosophies of the company, I want to tell you that this is also relevant to my subject of my topic of my discussion. These are the 3 guiding philosophies of the company we religiously follow. So it is very important slide I want to share with my younger colleagues, breaking stereotypes. So what we thought, I think some of the young people may not know the word what is stereotype. Stereotype means, it is an inbuilt thought process that, we keep it in our mind. Suppose like, mother-in-law, always, mother-in-law hates the daughter-in-law. It's a stereotype. It's a stereotypic thinking. So what we thought, we need to break the stereotypes in the way we do, in the way we think, in the way, we should not believe by being in India, we cannot do big things. That is what we thought. Every time, in every step I put forward, I thought breaking stereotype, no old thoughts. What people are thinking till today is not my thought. What we are going to think, from tomorrow, that is my thought. That is how we will move. This is one of the biggest, guiding philosophies of our company. And the second one is the road less travel. I like this particular phrase in English so much, the road less travel. That means if everybody is going in one direction, we should not go in that direction. We should go in the opposite direction, because wherever everybody is going, if you come to Tamil Nadu, see, that's where I'm coming from, in South India, there are, 1,000+ engineering colleges, and every kid in our household is studying engineering in the neighboring college. Actually, it is so unfortunate. So it really puts a stress on the educational system, as if there is no other course it is worth respecting. So what we should do the road less traveled. We should not go begin engineering. We should go opposite. We should go and study, veterinary science. You can go and study chemistry. You can go and study physics, and then you can become a space scientist. So I always adopted this second one very importantly. And the third is the most important, the guiding philosophy of DSK Innoscience, that is differentiating in science and contributing for the country. So these 2 words are the punch words for the DSK. We believe in everything we do. We differentiate in science. When we work with our collaborators, we don't work with Indian companies, because that is our business model. We work only with overseas companies. We have clients across the globe. We have clients in all over Europe, all over the United States. We have. So when we work with them, we always believe we will differentiate in science, and in the process, we will contribute to our country. That means country means India. So we will contribute to our country. We will give, we will give economy. We will give employment. We will give guidance. We will help our people wherever possible. There is so much we can do. So much we can do during our the recent COVID time. So much our company has done for the society, which I don't want to tell here because of the want of time. So this is our mission of our company. Basically, I don't want to take much of our time. We are driven by innovation and the difference in execution. This is what is our real guiding factor, and the respect for individual. In the company at DSK, the MD and the sweeping person all will be treated equally. We treat everybody equal, and then we operate the company. And I promised you that I will show you some pictures, and that is the picture here, long shot of our company. You can see we don't have a single village nearby, but we are in a highway. It is in a highway, and we are located in such a beautiful geography. And when I started the company, people used to really joke. But I can tell you about in the last 2 years during the pandemic period, all the companies in my neighboring city, that is Bangalore, they suffered with the pandemic. And I know in big companies, I can tell you today, more than 25 people died. They passed away. You know, during that peak pandemic time also, at DSK with 100 employees, we did not have single positive case. Single positive, we did 2 times RT-PCR test, no positive. Why? Because it is a beauty of the geography. We are in a very, very secluded geography. We do great science, but we are in a geography where it is such a beautiful oxygen around. The whole country is looking for oxygen, but we are completely soaked with oxygen. So that's how I thought we should move away from the bus of the people. And then we are not far away from Bangalore. We are 60 kilometers from Bangalore. And this slide shows 2 pictures, two pictures, and both are about the company. You can see the first picture where, you know, the company's main building is seen with an expansion on the first floor. And you see the second floor, you can see a brand new canteen built only about 6 months back, and it's catering all the employees. And in the middle, you can see a beautiful paddy field. Basically, since I'm coming from a farming background, you know, what we do, this is a beauty at DSK. I told many of my colleagues that this is the differentiator of DSK, and nobody in the country is doing it. And till now, nobody could tell that other company is doing it. If you have any company, you please let me know. I would like to learn. What we are doing in the company is about 5 acres of land. We grow vegetables and fruits. You know, for all the employees, our farm is full of vegetables and fruits. Our canteen is supplied by naturally grown vegetables and fruits, and we don't buy vegetables from outside. So that difference we made. We make some great healthy food, and right at DSK, all employees eat 3 times. So during pandemic period, I declare that the company, all the employees will take all the 3 meals in the company, and so that the possibility of, you know, interacting with the public is minimized. And we provide all the 3 meals free, and all the vegetables grown from our farm. And these 2 pictures will tell you a volume of whatever I said. And I don't know which company in the country is doing this, but we are doing it proudly, because I feel this is also another breaking stereotype. There is no need to think that we need a great lawn in front of us. I will do the science, you know, with abundant farming around. This is what we did actually. So this may be my last but one slide. Basically, this is the way our DSK would like to grow. This is my last year slide, but I would like to tell you very happily that many of the milestones we achieved within 1 year, which are shown in green. You know, a brand new 400 MHz NMR facility has been installed. This is the first 400 MHz NMR facility owned by a company in the state of Tamil Nadu. You know, there are a couple of other, private institutions and universities are holding NMR, but the first time we are having a 400 MHz NMR facility. We just installed about 4 or 5 months back. And our aspiration is to reach about 100 employees this year, which we already met. And we want to build an additional floor, we have done it. Food court, we have done it. So there are, you know, it's a kind of a fast growing company. So I think for the sake of time, maybe this slide I will skip it, but this is my last slide. So about this slide, I want to tell to all my colleagues, you know, very important message. You know, if you come and asked me about 4 years back, that company DSK, we were 25 employees and we were doing well. And if you ask me, Silva, you know, you have done well and there are 25 employees, are you happy? I would say, yes, we have done well, but our best is yet to come. And today, if you come and ask me, we have 100 employees, we have grown well, and we are doing well in business, and our collaborations are all going well. Today, if you ask me, Silva, today, are you happy? Yes, we are doing well, but our best is yet to come. Similarly, if you come to me after 7 years, and we do great work, a lot of expansion, we have 500 people, and if you ask me, Silva, how about you today? That day also I will tell our best is yet to come. I feel we should never feel we reach the best. There is always a better thing in this world than what others believe is the best that you have done yesterday. We have to keep improving. There is so much to do for the country. There is so much to do. I keep talking the word country with my employees so much. There is so much to do, and we have to do. We all have to do collectively. I see very bad when I see my people are living in poverty, and we all being educated people, we have to differentiate, we have to contribute, we have to give them the better life. This is how I think. So by having said that our best is yet to come, I think I am concluding. I don't know, I didn't keep my time. I don't know how I did, but please, all of you, please feel free to ask me questions. And also I'm available offline, whenever anybody has any question, they can approach me.
Thank you, Dr. Selva Kumar, for the really, really encouraging and scintillating lecture. It's not a lecture, actually. It is a kind of a personal journey. How did you come to a successful entrepreneur, transform yourself as an organic chemist, hardcore organic chemist and medicinal chemist, I could see, and becoming a successful entrepreneur and giving jobs to the hundreds of people sitting in the remote village of Hosur, not in a place where, it's in Bangalore. It's not a Bangalore or anything like that. It's a remote place where you have a personal experience. And I think you please start a, entrepreneurship development, training center in a DSK, sciences also. You're a fantastic speaker. I think there is no word to praise you, praise your experience, praise your thoughts, actually. So, see, this lecture is open for questioning. Is there any question?
I have a good number. Maybe I'll ask couple.
Sure.
Yeah, please.
Yeah.
Please.
So it was just really inspiring and mesmerizing. So I really enjoyed. I'm Madhu from IIT Kharagpur. I'm associate professor here, and I do organic synthesis, total synthesis, and I am a organic synthetic chemist by heart. Okay. And by coincidence, I also did my master from ISC Bangalore with Professor Govardhan Mehta. So then I moved to So yeah. So just Now I want to know, because as an academician, I also contribute in the industry. But what happened, we don't get a little entry or some information how I can contribute. Okay. So that is one question I have. Can we get guidance from you and how we can get it? And another very important questions I have, I used to ask several, I would say, representative from industry, how to know which are the, let's say, molecules we are importing from China. What are the APIs which, let's say, I want to make in kilograms in my laboratory, how I can make it? Because I am really working on it, but not getting proper leads or hints or connection. So these are the few things which always come to my mind. I would say I'm 39 years old and very younger than you, but I have aspiration to do this type of work. So I really inspiring. I would say you inspire a lot. It is really fantastic. So can you please comment on this point?
Yes, Madhu. Actually, it is very, very, very pertinent question from your side, and I am very happy to know that you are Govardhan Mehta's, student. Did you do master's there or PhD with him?
I did. I was integrated PhD, so I did master. Then I moved to Germany, and I stayed there for 7 years, 2 months for PhD and couple of postdocs. So then I came here, started my independent career about 7.5 years. So like about 4 PhD students about to graduate. So
Sure. I think it is very nice to see that, what I was mentioning in my presentation, that a faculty, is aspiring to do something outside their particular domain. So definitely see, since I'm interacting with a lot of professors overseas, they are how successfully they are starting the company, and then they are floating it. I know some of them what they do. Some of the best students, the students and the professors, they will be trying an idea, and then they will start a company. They will take a fund. They will take a fund of everything. Country to country, the funding mechanism changes, and then they will start something very small. They will incubate in the same lab, and I see they, in my first visit, I go to their lab and then discuss chemistry. In my second visit, I go to their new office and then I discuss chemistry. And then, when I go my third visit, they built 25 people, 30 people, like that. They started the company. The student yesterday has become the CEO of the company. The professor is still holding majority stake, and the student, the CEO, is also holding a big stake. Like that, I have seen, I would say, a number of companies overseas. And with them, we are all working actually. So with having said that, your question is like, how to, I also have a similar aspiration, and how to give a shape to that. How to give a shape to that.
Yes.
Basically, what we need to do, you address the point actually. We need to first address where is the requirement. Where is the requirement? That requirement is something that we have to go behind that, and then we have to develop it. There, what I believe is our development should be world class. Meaning, so see, coming from a pharma background, or I worked in Dr. Reddy's for 10 years or 9 years, I can tell you, for example, you take an API and there are several synthetic routes available. But what I do normally, I don't go to SciFinder. To be very frank, we have unlimited SciFinder in the company. I don't know even the password actually, because if I go to SciFinder, my mind is tied into the direction where everybody is going. So what I do, I sit and then write my own ideas. Sometimes I write 1 dozen ideas and give it to my chemist. Then they go on to study. They find 5, 6 of them are already there. Then we go behind the ones which is not there. So what we do normally, we always tell internationally, we have to develop an idea which is not reported elsewhere. It should be very attractive. It should be very quick. It should reduce the price of the product. Like that only we think actually. Like that only we think, and we believed, and then we moved, and we have been successful. Several times we have been successful actually. Some of the products and all, we saved a lot of money, and we could develop very, very attractive synthetic route, which has no literature precedent. So we don't work with literature. We are always going away from literature. So suppose if you are looking at an API product, which is selling at a higher price, and per kilo price is multiple lakh, I believe we can always reduce its price to 1/10 actually. Okay, if it is sold for 2,50,000, I can make the product in 25,000. Like that only I think. I don't simply think we will make 2,20,000. That is rubbish. Already it's selling for 250. Why should I go and compete? We should compete in a product. We bring the price to rock bottom actually. For that, what is required? You need your original thinking, and the molecule has to come in a very, very attractive and very, very fast way. Sometimes I think such products and all, the kind of ideas we put in, if we write a paper, we can write very good paper actually. But unfortunately, if I go to paper, that money we cannot make actually. So basically what I'm saying is you have to differentiate in the thinking process, and you have to come up with the excellent idea, synthetic route process for a very highly expensive product. So that market knowledge you should gather, and then you have to go behind that. This is only for the manufacturing I'm talking, but there is so much in the IP sector, in the intellectual property. You can generate IP, you can generate new knowledge, you can generate a new therapy, you can do so many things actually like that. So I think more than this, in the interest of time, Madhu, we can be in touch by email, and we can.
I'll be writing you the mail, and I'll be discussing more. I'm really interested to talk with you more. Thank you.
Yeah, thank you. Thank you, Dr. Maji. What Dr. Selvakumar told, in addition to that, there is a list of 38 key starting material requirement in India, which as Government of India has listed that we are totally depending on China. We can choose to produce any one of these key starting material, KSM, which is used for making API, and then we can start actually, we can become an entrepreneur. I think that's what I just wanted to add to what Dr. Selvakumar told. It's a fantastic one. Thank you. Thank you very much, Dr. Selvakumar, for the excellent lecture and very thought-provoking statements which you made, but still it's a fantastic one. Thank you. Thank you very much.
Thank you, Dr. Parsuram, for your excellent, appreciation, words of appreciation. Yeah.
Thank you.
Sure.
Thank you very much, Dr. Selvakumar. I feel proud of you and proud junior.
not junior, colleague.
Yeah, colleague. Really inspirational talk, and we wanted such a talk, and we have got the right person. And I hope you will speak even in future also, because we would like to hear you again and again. It is so inspirational, and so this is a great message to the younger generation.
I think you have to make him as a regular speaker in your RASAYAN year after.
Correct. You have spoken from your heart, and it has reached, or it has touched the heart of everybody, all the participants.
Sure.
And I wish all the best for your DS Sciences, highly innovative, really inspirational, and keep on inspiring us like this. Thank you very much. And Chirantan Rasayan Samstha is offering gold medal based upon your scientific, academic, as well as administrative achievements. So I hope you accept this. So this is the medal that is being offered to you, and this is the memento that will be offered to you, and also this is the third certificate. Okay, thank you very much.
Wonderful.
Congratulations for your achievement.
Thank you, Braja. I can tell you that, I can tell everyone, I have been a very silent performer all these days. And first time Braja has approached me, so I said, okay, our colleague is approaching. So I can tell you, I spent many days in making this presentation, maybe
Beautiful.
1 week, and each day, yeah.
Successful one.
So
Thank you.
This is the first time I have given this lecture, and the award you are giving me, it is the first award, and I will remember it forever.
Thank you very much. Thank you.
Great.
Okay.
We are also honored too. Thank
Sure.
you.
Okay. Bye.
Bye-bye. So, Mr. Pradhan, please. Prashant.
Okay, so thank you, Professor Selva Kumar, for concerning our thought on Indian and Western education and the need of celebrating failure just as we celebrating our success, and also how it is important to choose the road which is less traveled. Now we are in our final hour lecture, and we are eagerly waiting to hear the voice of Dr. Abhijit Mondal from Presidency University, Kolkata. I would like to request Professor Madhusudan Maji from IIT Kharagpur to kindly chair this session. So, Professor Maji, over to you.
Okay, thank you, Dr. Pradhan, and also I like to thank Chirantan Rasayan Samstha and Professor Bagh, particularly for giving me this opportunity to chair this session. And it was really mesmerizing from the morning, and one after another, a blockbuster talk, and it was really we are enjoying a lot. So now we are looking for the third talk of the morning. It will be delivered by Dr. Abhijit Mondal. So Abhijit is currently working as assistant professor at Department of Chemistry of Presidency University, Kolkata. Prior to joining the Presidency University in the Department of Chemistry, he has completed his PhD in 2016 from famous Indian Association for the Cultivation of Sciences under the guidance of Professor Nikhil Ranjan Jana. And after completing his PhD, he also did his postdoctoral research from Republic of Korea on 2D based nanomaterials for electrochemical water splitting reactions. And today, Dr. Mondal will be presenting on with the title Potentiality of Nanomaterials as Biosensor Probe and Electrocatalyst. So I'll not go much details about his talk because his abstract is already available. So I'll rather look forward to his talk. So I request Dr. Abhijit Mondal to start presentation.
So thank you, Professor Madhusudan Maji, for a nice.
So can you please share your slide? And yeah.
Okay. So is it visible?
Yeah, perfect. Please continue.
Oh, okay. So thank you, Professor Madhusudan Maji, for a nice introduction. And first of all, thank you to CRS organizing committee and members for giving me this opportunity to represent my work entitled Potentiality of Nanomaterial as Biosensor Probe and Electrocatalyst. So nowadays, nanomaterials are hugely applied in various fields. So in this first slide, I have shown that nanomaterials are applied in various applications, just like water splitting, fuel cell catalysis, optical sensing, electrochemical sensing, and other application, just water purification, battery-based application, photocatalysis, and supercapacitor. And there are many other applications. So mainly I am focusing the nanomaterials like plasmonic nanomaterials like silver, gold, or catalytically active nanomaterials like palladium, platinum, and the nanomaterials which contains high surface area, high conductivity, as well as the catalytic activity, just like graphene or reduced graphene oxide and nickel sulfide. So we know that what are the nanomaterials or the nanoparticles. The materials which are in nano dimension. So we can say by a simple analogy that 1 rupee in 1 crore, that is the nano dimension. So due to this nano dimension, it has some typical characteristic. Due to this typical characteristics, so it is applicable for different fields in research. So there are some typical features like compared to bulk material, like nanomaterials have large surface area, high catalytic activity, and some nanomaterials shows plasmonic activity, as well as high conductivity and fluorescence quenching activity, etc. So I have shown in this first slide some TM images of the nanomaterials or the nanocomposites. So this first image, TM image, shows that it is the image of beta cyclohexene graphene hybrid, where the graphene actually it is actually reduced graphene oxide. So reduced graphene oxide is stabilized by beta cyclohexene. Beta cyclohexene is a cyclic oligosaccharide, so it has a hydrophobic cavity and outer surface is hydrophilic in nature. So, and the reduced graphene oxide has the property of fluorescence quenching, and the beta cyclohexene has the recognition ability. So we can use those both property, and we can sense the important biomolecules like cholesterol. And there are the nanomaterials like different composite on graphene based. So gold graphene, where the gold nanoparticles around the average size 4 nanometer, which is distributed on the edges of the reduced graphene oxide. And the for palladium graphene nanocomposite, the palladium nanoparticle with average size around 3.4 nanometer are distributed on reduced graphene oxide surface. And for particularly platinum graphene, where the platinum nanoparticle are distributed throughout the graphene oxide surface uniformly. And this nanocomposite is shows good performance in fuel cell catalysis. And there are the nanomaterials like tungsten metal sulfide, just for you can say nickel sulfide. So nickel sulfide has the morphology like 2D, two-dimensional flex-like morphology. So it has high surface area, as well as it shows the activity towards the electrochemical water splitting. And nowadays also the green synthesis is very important to prepare plasmonic type nanomaterials, just like gold nanoparticle or silver nanoparticle. And we can use this plasmonic nanoparticle for biosensing purpose, or it has the antibacterial property. So now we are coming to the beta cyclohexene hybrid for cholesterol sensing. So we have used a simple strategy for sensing of cholesterol. So if the beta cyclohexene graphene hybrid solution is incubated with rhodamine 6G like dye, so then here the reduced graphene oxide surface used as a fluorescence quencher. So rhodamine 6G dye, it will go to the hydrophobic cavity of the beta cyclohexene, and the distance between the dye molecule and the reduced graphene oxide surface is decreased. So there is a fluorescence energy transfer. So the fluorescent intensity of the rhodamine 6G dye is quenched. And if we add the cholesterol molecule, then the cholesterol molecule has higher binding affinity towards the cavity of the cyclohexene. So it replaces the rhodamine 6G dye, and then fluorescence is turned on. So fluorescence is turned on. So using this simple strategy, we can we can detect the cholesterol with different concentration, and we can go to the detection limit around 5 nanomolar. And if we add the cholesterol cholesterol concentration about 20 micromolar, so we can see visually that the fluorescence is turned on. So this image actually taken in normal UV light. And this simple strategy is used for the detection of the cholesterol present in blood serum. So this serum actually, this normal concentration of cholesterol containing blood serum. So overall, the process is very simple and cost effective. And we know that the graphene has high surface area as well as high conductivity, and the nanoparticles like noble nanoparticles or the gold nanoparticle has catalytic activity as well as plasmonic activity. So you can use the catalytic activity of gold nanoparticle and the conductivity of graphene, reduced graphene oxide surface, reduced graphene oxide. So we can couple both property and prepare nanocomposite that is used for the electrocatalytic sensing for different biomolecules in these three molecules, and the concentration of ascorbic acid is naturally high compared to dopamine and uric acid. So generally it suppresses the peak of the oxidation peak of the dopamine and uric acid. So it is very important to separate this peak. So by using this gold graphene nanocomposite material, catalyst, we can selectively detect ascorbic acid, dopamine, and uric acid. Whether simply graphene modified electrode or gold modified electrode or bare electrode cannot able to sense selectively. And this modified electrode is used for sensitive detection for DNA to DNA bases. DNA bases like guanine, adenine, thymine, and cytosine. And if we simply perform electrochemistry using uric acid by this modified electrode, so with increasing the concentration of uric acid, that there is current is linearly increasing with the concentration of uric acid. So we can get a standard curve, and we have taken the blood serum sample from hospital, and that we have taken the two sample that is contain, one is a normal concentration of uric acid, and another is high concentration of uric acid. So from using the blood serum, we can get the we can perform we have performed the electrochemistry, and we get this current intensity, and this from this current intensity we can get the unknown concentration of uric acid in blood serum. So now the gold graphene composite actually shows the catalytic activity. So we can prepare a noble metal graphene composite in general type. So this nanocomposite can be applied for fuel cell catalysis. So for that purpose, we have developed a simple strategy for synthesis of noble metal graphene nanocomposite using partially reduced graphene oxide and followed by addition of metal oxide, noble metal oxide, hydroxide nanoparticle solution, and then there is a redox reaction between them. So we can get the noble metal graphene nanocomposite. So redox reaction is occurring because the standard reduction potential of noble metal oxide or hydroxide nanoparticle is very much high compared to the partially reduced graphene oxide. So that's why we get the noble metal graphene nanocomposite, and particularly the platinum graphene nanocomposite, as I have shown the TM image in first slide. So the nanoparticles are uniformly distributed throughout the graphene surface. So for this characteristic, actually platinum graphene nanocomposite shows great performance towards the electrooxidation of formic acid and ethanol, and also it shows the high stability under 100 cycle compared to control catalyst. And this image actually shows the bar diagram for ethanol electrooxidation. So here the platinum graphene as well as palladium graphene also shows good performance, but for palladium graphene case, actually the stability is lost with time. And this the beautiful feature of this composite is free from any surfactant. because if the surfactant is present, then it will cover some active region of the nanoparticle. So in that sense, this nanocomposite is very important for this type of application. And now now actually the requirement of green energy through green process is very important. So there is a way that we can get hydrogen as a green energy by electrochemical water splitting. There is another two way we can get by photocatalytic water splitting or photoelectrochemical water splitting. So now we are discussing about the electrochemical water splitting. So by electrochemical water splitting, we can get the hydrogen as a green energy through green process. So a water splitting actually contain two consecutive reaction. One is oxygen evolution reaction, another is hydrogen evolution reaction. So for the oxygen evolution reaction, it actually involves 4 number of electrons. So this process is kinetically more slow process compared to hydrogen evolution reaction. So it is very important to develop catalyst for the oxygen evolution reaction. So for that purpose, we have taken the pencil graphite act as an electrode. So we have taken pencil graphite, which is commonly used as for writing purpose. So this pencil graphite is modified by nickel sulfide by in situ process, and this electrode act as a anode in a 3 electrode system where platinum wire act as a cathode, and silver chloride act as a reference electrode. So this nickel sulfide modified this pencil graphite is shows great performance in oxygen evolution reaction. We studied the linear sweep voltammetry by electrochemistry. So this linear sweep voltammetry shows that the NiS2 modified pencil graphite shows great performance compared to standard catalyst like iridium oxide. And from Tafel slope, the slope is less compared to iridium oxide. So this actually corresponds to the faster charge transfer between the molecule, between the analyte molecule and the electrode. So due to this, the nickel sulfide modified this electrode anode is shows good performance. And the one possible mechanism that for this good performance is due to the shape of the electrode. Actually, if we compare with the glassy carbon electrode, so glassy carbon electrode contain flat surface. So if the bubbles form on the flat surface, then it will generally stack on the flat surface for some time, and with time actually bubbles grows. With time bubbles grows. So it prevents between the connection between the electrode surface and the newer electrolyte. So there is a diffusion problem. So for that purpose, we have taken the cone-shaped electrode. This cone-shaped electrode, here the red circle actually represent the bubble. So if we go down from upside to bottom, then the number of bubbles actually decreasing. The number of bubbles are decreasing. So and also in the same time, the depth of the solution is increasing. So bubbles formed at the tip is in high pressure. Difference. So so the bubbles form at the tip, it will move through the solution. Either it will move through the solution or it will move through the electrode wall. So when it moves to the electrode wall, it definitely applies some pressure or force on the bubble next to it. So in this way, they move. The bubbles are moving upward and they are accumulate on the Teflon coated surface. So here you can see the bigger bubbles on the Teflon coated surface, not in the active region of the electrode. So there is no diffusion problem, and we have tested the stability using this anode. So we can get the stability up to 25 hours. We have tested up to 25 hours, and there is no current loss. So so that's why this electrode actually shows the good performance. So now we are interested to modify further this electrode for hydrogen evolution reaction. And as I have said, that the green synthesis technology is very important for synthesis of plasmonic nanoparticle, for sensing of heavy metal ions, or it has some antibacterial property. So for that purpose, we have synthesized the silver nanoparticle using garlic extract and ambient light source. So using this silver nanoparticle, we can detect the marker ion, but it is not new thing. By using the plasmonic nanoparticle, anyone can sense the mercury ions. But if we add iron on the silver nanoparticle solution, then there is a redox reaction between silver nanoparticle and Fe3+, and it results that the color of the silver nanoparticle solution is ultimately decolorized and form colorless solution. So there is obviously there is a redox reaction between silver nanoparticle and Fe3+ ion. And in general, the concentration of Fe3+ ion in important. So it is very important to suppress the effect of Fe3+ ion. So for that purpose, we have taken silver nanoparticle in PBS phosphate buffer medium. For that purpose, we have taken the silver nanoparticle solution in PBS buffer medium, and there is a well-known chemistry between the Fe3+ ion and phosphate ion, because there is a complex formation, and that complex will not take part in redox reaction. So we can suppress the effect of Fe3+ ion, and then we can detect the mercury by using this silver nanoparticle up to 2 micromolar level. And here you can see the UV visible spectra, and with increasing the concentration of mercury, the surface plasmon resonance is decreasing. And also this strategy is quite selective against other biomolecule or ions. And now to increase the sensitivity, because here the sensitivity is 2 micromolar, to increase the sensitivity further, we have further reduced the silver nanoparticle, because we can expect that the silver nanoparticle is prepared by natural ligand. So we can expect that the silver nanoparticle is not completely reduced. So that's why we have reduced the silver nanoparticle by adding the ascorbic acid. And
Dr. Mondal, so you have to,
Hello?
Yeah, can you hear me? So time is also, we are running out. So please,
Okay, this is the last slide.
Yeah, okay.
This is the last slide. Okay. So if we add the ascorbic acid, then the depth of the color of the solution is increasing, as well as the UV absorption intensity also increases. And we can get the indirect proof from cyclic voltammetry study, that if we perform the cyclic voltammetry using these 2 nanoparticles, silver nanoparticle and further reduced silver nanoparticle, this is the red color shows. So this black color curve shows that the reduction peak area is higher compared to the red one. So this corresponds that this signifies indirectly that the silver nanoparticle, which is prepared naturally by natural ligand, is not completely reduced. So using this silver nanoparticle, we have detected or enhanced the sensitivity of mercury ions up to 100 nanomolar. And here we use the absorption minima instead of absorption maxima. So here using the absorption minima, we detected the mercury ions up to 100 nanomolar. And then using the silver nanoparticle, which is prepared using the garlic extract, is used for the sensing of cadmium ion. So cadmium ion actually it forms complex with the ligand present on the nanoparticle surface, and at a certain concentration, around greater than equal to 12 micromolar cadmium ion, it will form the complex with the nanoparticle, and with time it will precipitate out from the solution. So using that strategy, we have detected the cadmium ion using the silver nanoparticle. And now we are interested to modify the glass substrate, or we have to study the further for this absorption minima. And we are interested to modify the glass substrate using this silver nanoparticle and used for sensing of mercury or lead or cadmium in practical purpose. So thank you again for your kind attention.
Okay, thank you, Dr. Mondal, for your insightful talk about on nanomaterials and their applications in sensing and water splitting reactions. So now we can have quick 1 or 2 questions if there.
Sir, I have one question.
Yeah, Argo,
Okay.
can you please? Yeah.
Yes, sir, myself Argo from Vidyasagar University. Sir, I have a very basic question. At the beginning of your presentation, you enlightened a word, plasmonic nanoparticle. Sir, I want to know what is actually plasmonic nanoparticle. Is it related to surface plasmon resonance?
Yes. It is related to surface. Surface electrons are oscillating. With
So
the
if you could briefly,
actually, these are the inherent property of the nanoparticle or nanomaterial. So generally, the silver and gold nanoparticles are showing the plasmonic behavior. So the surface electrons are for that nanoparticle. Surface electrons are loosely bounded, so it will oscillate. And if you irradiate with the light, then a specific light is absorbed. So and corresponding light is actually you will see. So for that reason, you will see the different color. Actually, you can see the red color or yellow color based on the specific light. Actually, the nanoparticle is absorbing due to this surface plasmon resonance.
Thank you, sir. Thank you very much.
Okay, thank you. Either one more question maybe we can take? Quick question if it is there.
Okay.
Which not the case, as we are also running out of time. I thank you, Dr. Mondal, for your nice talk, and I hand over to Professor Bach for conferring the award.
So thank you very much, Dr. Mondal, for your excellent presentation and excellent work. So the society, based upon your achievements, offers you this CRS bronze medal and also this memento, and please accept this. Thank you very much. Very nice
Thank
work.
you. Okay.
Yeah.
Oh,
and also
accepting.
the certificate. This certificate. Okay. Thank you very much. Very nice work.
Thank you. Thank you very much.
Okay. Thank you.
Mr. Prashant, please.
Thank you, Dr. Mondal, for your insightful presentation. Now we are going to begin our poster session, and which will be chaired by Professor Prasanjit Mal, NIACR Bhubaneswar, and Professor Madhusudan Maji. So over to you, Professor Mal and Professor Maji.
So thank you, CRS, for giving me this opportunity to chair this session, and I would like to thank Dr. Madhusudan Maji for agreeing this session. Okay, so now we'll start the poster presentation. I think there will be around 10 posters. So the first actually is the serial has been given, or
yeah. It will be P1 to P10. P1 to P10.
Okay, P1 to P10. Okay. So
P1, and if he misses, then we'll go to the P2.
Okay. I hope that all the
poster presentation will be for 5 minutes in total.
And any question answer? Any question answer?
Yeah, including that 5 minutes.
Okay. So now I'd like to request Dr. Madhusudan Maji to formally start the session.
Okay, I think let us thank you, Professor Mal. So let us go by poster 1, and as already mentioned by Professor Bach, P1. If it is there, whatever I have poster abstract, it should be Mahmudul Hassan, right?
Yeah, Mahmoudul Hassan. Yeah.
So can you please start your presentation?
Okay, okay.
Mahmoudul Hassan currently doing a PhD, right?
Yes, sir. I am under
Yeah,
Dr.
CBMR Lucknow under the guidance of Professor Buddhadev Chattopadhyay. So there, it's a nice chemistry. I think let us directly listen from Mahmoudul. Can you please start, Mahmoudul, your presentation? Yeah.
No award, right? Only the PhD people will listen.
Yeah. Yeah, Mahmoudul, can you please start sharing your screen?
Yes, sir. I'm sharing.
And P2 be ready.
And P2, it will be Professor, Dr. Mr. Bera, again from CBMR Lucknow. Please be ready, P2.
Is it visible, sir?
It's coming. Yeah.
Very nice. Yeah, please. Go ahead.
Thank you. Thank you to Professor Mal and Maji for chairing this session, and I'd like to thank the Chirantan Rasayan Sanstha for giving me this opportunity to present my research work. I am Mirza Muhammad Mahmoudul Hassan, working under Dr. Buddhadev Chattopadhyay in CBMR Lucknow. My research work entitled as Remarkably Efficient Iridium Catalyst for Directed CSP2H and CSP3H borylations of diverse classes of substrates. In this work, we have developed two catalysts, first generation CB1 catalyst and CB2 catalyst from these two newly designed two pyridyl thiophene ligand. They are utilizing the acidic photon. Sorry.
Can you please zoom it because nothing is visible, right?
Yes.
So whichever you want to talk, please zoom that particular area and then slightly more. Yeah. Now go to that particular place and yeah.
Yes.
Okay.
These two catalysts CB1 and CB2 from these two pyridyl thiophene ligand using this acidic CH bond, and we among these two catalysts, CB2 is the air stable, and we develop CH borylation condition under atmospheric condition, and with these two catalytic systems CB1 CB2 we have developed ortho CH borylation of various arene system of more than 35 class of different functional group, including aliphatic, heterocyclic, as well as late stage borylation, and these two catalysts were confirmed by their exact crystal structure. And we optimize the reaction condition using these two catalysts, CB1 and CB2, and also with ligand system, and our optimized reaction condition gave 95% isolated yield with this aromatic amide and 99% ortho selectivity. With this optimized reaction condition, we explore the substrate scope with different arene system, and I have shown here some selected example, and this arene system have more than 35 class of different functional group, and this shows same reactivity towards the orthoborylation of arene. And I have seen here some selected example as well as heterocycle borylation, and we did late stage borylation of this heterocycle molecule, and it gives excellent selectivity with 77% yield. And we also have aliphatic borylation with this two pyridyl substrate, and here we got new selectivity with this methyl was borylated without touching this benzylic position. Even with the late stage borylation of this peroxetine drug molecule, and it gives 58% yield, and it gives borylation at the less crowded position without touching the other position as well as this ring. As our catalyst, this CB2 catalyst is air stable, we developed a condition where borylation reaction happen under atmospheric condition, and there is no need of inert gas atmosphere. As in iridium catalyzed borylation reaction, there need of inert atmosphere condition, and we develop reaction under atmospheric condition, and it gives excellent outcome with these substrates. These are selected substrates, and if we go to the mechanism, as you know, iridium catalyzed borylation goes via the iridium teeth boryl complex formation. But when we started the reaction mechanism with the CB1 catalyst adding B2B2, we got quad-ligated bis boryl complex, and it is confirmed by NMR spectra. And after concomitant loss of quad 1,5-cyclooctadiene, we got iridium bis boryl complex, and with this vacant site, and this intermediate also we detected in the HRMS. So this active intermediate iridium bis boryl complex, where after addition with substrate and after oxidative addition and reactive elimination give the product, and from this intermediate with B2B2 regenerate the catalytic cycle. In conclusion, we have developed two newly designed catalysts, and with this catalytic system we borylate more than 35 different classes of substrate, including aliphatic, heterocyclic, and late-stage borylation. And this work has been published in JACS and highlighted in Science. And last, we gratefully acknowledge the UGC, DST, CRB for financial support and Director of CBMR. Thank you.
Okay, so Madhu, please.
Yeah, thank you. So if there is one question, is there any question? If it is not, then let me ask one question. So is there, you found any ketone reduction or something like this? Like say,
No,
this example, benzophenone you are doing?
Yeah. We have done benzophenone, acetophenone system, and in this reaction condition, the ketone group is not reduced. And this is a very interesting case.
Yeah, that is what I also got little bit. It's very interesting result, and also very selective. Then really nice. Also, acetophenone also work, right?
Yes, acetophenone also work. It is not reduced.
Okay, thank you. So thank you, Mr. Hassan, for your excellent work and presentation. So let us move for the next poster. So, Professor Mal.
Yes.
Yeah. Let us.
Yeah, let's start now. We'd like to invite Mr. Bera for his just to start his presentation. Okay, so he should be given 5 minutes. Mr. Bera, please.
Yes, sir. Is it visible?
Nothing is visible. Please share your content.
Visible?
Not yet.
Okay.
Have you shared your slides?
Yes, sir.
Please, there are 3 dots and go there and just now it's coming.
It's coming. Very good. Yeah.
Yeah, how do you pronounce your name? Just sorry to interrupt you.
Pishosomo Bera.
Pishosomo.
Okay, very nice. Okay. Please zoom and describe.
Good morning, everyone. Myself, Pishosomo Bera. I am working as a PhD fellow in CBMR Lucknow. Today, I will present a poster, Rapid Assembly of Functionation Morpholin on 4-oxapine via 3+3 and 3+4 annulation of azoxyl cation and amphoteric compound.
Can you please zoom a bit because it's not visible? Can you please zoom?
Yes.
Because it is not visible. As Professor Mal already mentioned earlier.
Yeah. So please zoom it.
Yes. Is it visible?
No, not. I think your net is slow. So let's wait for. So make it, if it is 30%, make it 100%, something like that. Make a right click and zoom it.
Is it visible?
No. And see, whenever it is the poster presentation, you could have made it a 5 minutes, 5 slides. That would have been much better actually.
Yes, sir.
Yeah.
So can you take the help of others that how to zoom it, because zoom options should be there.
The bar is showing less. I think.
Yeah. So
No, you can go down. There is 14% only it is.
so make it 100
Left down.
200%.
No, next.
All right.
Or through little bit right.
So it is a 14%. Make it 50.
That for plus sign you just press on a bit.
That's it. Right. Yes.
Yeah.
Some more.
Okay. This is fine. Very good.
Now go to the appropriate places.
Yes. First, I will talk about azoxyl cation. So before 2011, azoxyl cation had been suggested as an intermediate in some process. For example, from the reaction of alpha halomide with base and the isomerization of alpha lactam and oxygenating. But the existence of the azoxyl cation has never been proven experimentally. But in 2011, Jeffrey group first trapped this azoxyl cation. After that, several groups started work with azoxyl cation. But our strategy to synthesize saturated N-heterocycles from azoxyl cation, some popular group, Agoral and Carrier group, they also reported saturated N-heterocycles in different process using vinyl sulfonium salt and fused oxygen and SNAP reagent. So from this work, we wonder if we could use this azoxyl cation to synthesize saturated N-heterocycles that is important in drug discovery. So to develop this reaction, we needed to engage nucleophilic heteroatom and electrophilic carbon center is required. So actually nucleophilic heteroatom and electrophilic center are separated some number of certain atom. They are not connected mesomerically. Actually we need amphoteric system. So here some challenges to develop this reaction, because here olefin group and keto group are present. They are known to react with azoxyl cation. So with this challenge, we started the model reaction with the azoxyl cation and hydroxyanone. We got best result in presence of sodium carbonate in HYP. So with optimization table in hand, then we explored the substrate scope with different 1,3 amphoteric system. Next we also after that then we changes hydroxyanone to hydroxyalkynone system. Here we got jet selective product. After that we noticed an interesting observation. When we kept the sample in NMR tube, then we recorded NMR data, then the vinylic photon shifted from the previous one. That time NOE study suggests that E isomer is formed. So basically Z isomer is formed initially, then it is kinetic control product, then it is transformed to E isomer, that is the thermodynamics control product. Then we also deprotected benzyloxy group from Z and E. We also get Z isomer due to intermolecular hydrogen bond. So after that 3+3 cycloaddition reaction, we also focus on 3+4 cycloaddition reaction with different 1,4 amphoteric system. Here also another amphoteric system that is structurally different from previous one. Here olefin group is terminal position. Here we first got uncyclic product, then it was cyclized in presence of triethylamine. And another amphoteric system 12 that is also structurally different from previous 7. And here we got 3+2 cycloaddition product, but we did not get any 3+4 cycloaddition product. After that, we also explored the follow-up studies. Here, hydrogenalysis of the morpholine, we got product 15, and it was cyclized under Mitsunobu condition. We also deprotected benzyloxy group from the 7-member ring, and we get 3 amine, and then it was cyclized between the free amine and aromatic bromide. So that's it, and our paper is published in JOC, and our paper is highlighted in SynFact. So thank you.
Okay, thank you, Mr. Vega. So for this nice presentation, I would like to request the audience if they have any questions, they can basically interact with him. Questions, please. So I have just a kind of small query about this isomerization. You said that what is the driving force with the isomerization? I just missed that particular point.
Isomerization, that is actually in presence of acid or PTS acid. In presence of acid, this vinyl oxygen center of the vinyl amide protonation due to push-pull effect of nitrogen, and this double bond act as single bond character and rotate this double bond, and it's formed to transform Z to E isomer.
Okay, so that is the only just simply acid catalyzed isomerization. Okay, okay, I just, because that is coming from your CDCl3 or what?
We kept the sample in CDCl3 overnight, then we recorded the NMR data, then vinyl proton is totally.
No, did you check if you just use the kind of acid-free CDCl3?
No, yes, we check also in CD2Cl2, then it was not shifted.
All right, okay.
Which one is more stable? Do you think that one is more stable, the one that you are getting after putting NMR?
Yes, E isomer is stable. We also collaborate DFT calculation. Then DFT calculation also suggests that E isomer is thermodynamically stable product and Z isomer is kinetic control product.
Okay, very good, very good.
Yes.
Okay, so now I would like to give thanks to Mr. Bera for his excellent presentation.
Thank you, sir.
Now the poster 3, I'd just like to request Mr. Somnath Pulobi, right?
Yes, sir. Yes, sir.
Yeah. So please start your presentation.
Okay, sir.
Please zoom it some more.
Yes, sir. Is it visible, sir, clearly?
Some more.
Okay. Is it okay, sir?
No, it's not okay.
Okay, sir.
Zoom in.
Okay, sir. It is okay, sir?
No, it's not okay. Why don't you? There are options. Why don't you go to 150? Yeah. Okay, now it's okay.
Yes, sir.
Yeah, please go to the portions where do you want to discuss. That's it.
Yes. Now I am Somnath Pulobi from Vidyasagar University. I am working in the photocatalytic behavior of silver-ion coordination polymer under the supervision of Professor Sudipto Dalai. And now I have selected my research topic as silver metal because silver metals are very much photosensitive. Because on irradiation to UV light, there occurs a facile transfer of electron from conduction band to valence band. And in the present situation, in the textile industry, there are methylene blue is generally used for coloring the textile. So these water that are released from the textile industries are generally mixed with reverse ponds etc., which polluted the water. So these polluted water are very much harmful for the aquatic animals and also the human being. So I want to develop a photocatalyst which can degrade this methylene blue to colorless portion that is not toxic. So for this purpose, I have developed two coordination polymer of silver, mainly that is prepared from the aromatic carboxylates. Aromatic carboxylates that are derived from the trimesic acid or 1,3-benzene diacetic acid. Now why I have selected the aromatic carboxylates, because during this process, a transfer of electron from valence bond to conduction band is necessary. So, if the aromatic moiety is present in the system, that will help us to facile towards facile electron transfer. So this excess structure of the two coordination polymers are highlighted below. Here, in both the compounds, the metal ions are connected by bridging dipeptidyl ligands, and the carboxylate anions, aromatic carboxylate anions, bridge these two chains, and that results in the generation of one-dimensional coordination polymer. And now the difference between the two structures is that in the compound 1, only AG-BP chains are present and aromatic carboxylates which link the two AG-BP chain. But in the structure 2, the aromatic carboxylate that is derived from the trimestic acid participate in the coordination compound formation. Now I have measured the band gap energy. Now I have measured the band gap energy of the two coordination polymer for their photocatalytic behavior. The band gap energies are 4.47 eV for 1 and 4.50 for 2. Because at the band gap energy in the range of 4.5, so when UV light is irradiated in the complex 1 and complex 2, there occurs a transfer of electron from the valence band to conduction band, and their photocatalytic behavior of the coordination complexes are studied in the aqueous methylene blue solution in the UV spectrophotometry by gradual decreasing of their UV absorption data indicates that with the propagation of time, there occurs degradation of methylene blue. Although the time takes 190 minutes, but the degradation is 94%. Degradation is 94% by the coordination compound 1 with rate constant 14.04 into 10 to the power minus 3 minute inverse. And for the coordination complex 2, it is 10.43 into 10 to the power minus 3 minute inverse. There, the linear correlation between C by C0 and time inverse indicate it is a first order rate equation, and there occurs 94% degradation by the coordination compound 1. And here the mechanism for the degradation of methylene blue has been studied by us, and it is established that when UV light is irradiated, there occurs a transfer of electron from valence band to conduction band, and this will generate a hole in the valence band. This hole captures one electron from the water and convert it to hydroxyl radical, and also the oxygen molecule present in water also might accept one electron to produce O2 dot radical. This will also convert it to HO dot, and this as a whole degraded the methylene blue, and the color of the methylene blue intensely blue, but after the degradation, that is with the propagation of time, it is described here. With the propagation of time, the UV intensity absorption peak gradually decreases. So we may say that at the 190 minute time, the solution become colorless. So there occurs total degradation of methylene blue. Again, this photostability of the catalyst is justified by the PXRD experiment and also the IR study. PXRD experiment, IR study indicate that the photocatalyst remain as before during the photocatalytic process. And here, now the question is why compound 1 is far better towards the photocatalytic behavior compared to compound 2. In the compound 1, there occurs their presence of high aromatic moiety, that is the presence of 1,3,5 tricarboxylate anion occurs a facile transfer of electron from valence band to conduction band. And here in this process we have already known that silver metal is a detained system. So on irradiation of light, there occur transfer of electrons. So in our future plan, we want to work on the generation of photoelectric current on the silver coordination polymer. Thanks everybody.
Okay, so it's a nice work, and the questions from the audience now. So no questions, then I'll ask one. So what is the basically irradiation wavelength that we use for the excitation of the silver valence band to conduction band?
Sir, it depends on the band gap energy. So we have generally used 250 watt high pressure
No,
mercury
what's the wavelength of the UV radiation you used?
near about 270 to 275 to 300.
Is the UV irradiation?
Yes.
So which instrument did you use?
We have studied the absorption peak measurement in the UV instrument, UV spectrophotometrically, and we have a setup to irradiate. We have a setup in our lab to irradiate the complete.
Okay. I think you did not get my point. So anyway, I'll switch over to Professor Bagh to ask the question now.
No, I think your question matches with my question. Like, where from did you measure this cyclic voltmeter or this redox? In which instrument? This is what?
Sorry, we have not worked on the generation of photoelectricity because it is our in the future plan. We have now studied on the photo degradation.
No, this particular plot, where from did you get this on this energy electron volt versus this one?
Torque plots.
Yeah, yeah.
Torque plot.
This is in which instrument? This is what I meant.
Torque plot generally done after you have to done the UV measurement of the complex one. Then from the UV data, you can easily plot the torque plot. First, you have to measure the UV data from the instrument. Then you have to plot the torque plot.
Okay. All right. Thank you.
Okay, so thank you. And now I'll have to request Dr. Maji to introduce the next speaker.
Okay, so yeah. So next speaker is I think Mr. Dinabandhu Sasmal. He's working under Professor Tridip Tripathi from Midnapore College, Autonomous. So I request Dinabandhu, please start your presentation. Share your screen and start your presentation. Poster number 4. Dinabandhu, are you there?
Am I audible? Hello.
Yeah.
Yes.
Sir, am I audible?
Yes,
you're audible. Can you please share the slide?
Is it visible?
Yes.
Thank you to Chirantan Rasayan Shastra and everyone to give such opportunity to present my research work. Myself, Dinabandhu Sasmal, I am doing my PhD work under the guidance of Dr. Tridip Tripathi, Midnapore College, Autonomous. My topic is study of cadmium-2 ion removal from its aqueous solution by amylopectin graft polymethacrylate co-sodium acrylate. Among the potentially toxic metal ions, cadmium-2+ ion is very much toxic in nature. Present study deals with the cadmium-2 ion adsorption by using biodegradable copolymer amylopectin graft polymethacrylate co-sodium acrylate, which is a new class of easily available adsorbents. First, amylopectin graft polymethacrylate was synthesized by using the materials like amylopectin methacrylate, potassium perdisulfate by radical polymerization technique. Then the product was hydrolyzed by sodium hydroxide solution to get amylopectin graft polymethacrylate co-sodium acrylate. Now, the characterization techniques are as below. New peaks appear in the proton NMR plot, which are corresponding to delta 2.09 for methyl groups and 2.01 for CH groups and 1.43 for methylene groups. And in the FTIR spectra, 1635 centimeter inverse and 1465 centimeter inverse stretching frequencies are for CO bond of COOMe and carboxylate ion respectively. And the granular structure of the amylopectin has drastically changed in the same image. And TGA plot also confirms the formation of the graft copolymer. Cadmium-2+ ion adsorption study was done by the following way by using this equation. The effect of pH, effect of polymer dose, contact time and temperature to the adsorptions are plotted here to find out the optimum conditions for the adsorption process. The optimization conditions are pH 5.5, polymer dose 0.02 gram, and 120 time has been taken, and temperature at 90 degrees Celsius. From the R-square values, it is clear that the adsorption mechanism follows pseudo second order rate equations because R-square values are close to 1. And the absorption data are well fitted with the Langmuir adsorption model having a QM value 160.742. And the conclusion is amylopectin graft polymethacrylate co-sodium acrylate was prepared successfully by first grafting polymethacrylate chain onto the amylopectin backbone by using potassium perdisulfate as the initiator, followed by alkaline hydrolysis, and the graft copolymer was an efficient cadmium 2 ion adsorber from its aqueous solutions, and the kinetics of the adsorption was well explained by the pseudo second order kinetic model, and the substance data were found to obey the Langmuir isotherm model. Thank you.
Thank you, Mr. Shashmal, for your talk on cadmium ion removal. So, if there are any question? Is there any question? Okay, so if this is not the case, so I request. So, Prosenjita, should I ask for the next?
Yeah, please call the next. Yeah.
Okay, so next, thank you, Mr. Shashmal, for your talk, and you may just unshare the screen. Now we request poster number 5, P5. So Siddhartha Das, Mr. Siddhartha Das, who is currently working under supervision of Professor Shumita Roy, Department of Chemistry, Vidyasagar University. So Siddhartha, can you please start your presentation after sharing?
Yes, yes, sir. Yes, sir. Is it visible, sir?
Yeah. Okay.
Myself, Siddhartha Das.
This is okay. Can you increase the font, because the writing is so much and nothing can be read. Yeah. Continue, please.
Myself, Siddhartha Das. I am a PhD scholar of Department of Chemistry, Vidyasagar University, under supervision of Dr. Sumita Roy. I like to present on the topic 6-acyl amino nicotinic acid based hydrogylator applicables in phase selective gelator, reproducible mat formation, and toxic dye removal. For this purpose, 3 amphiphils named as 6-octanoyl amino nicotinic acid OANA, 6-dodecanoyl amino nicotinic acid, and 6-dodecanoyl amino nicotinic acid were synthesized. This is the synthesis scheme of the amphiphils. This is the energy minimized structure of the amphiphils. The critical chain length of OANA is 1.42 nanometer, and DANA is 1.67 nanometer, and DDANA is 1.93 nanometer. At first, we investigated the self-assembly properties of the synthesized amphiphil in aqueous solution. For this purpose, all amphiphil were transformed in sodium, their corresponding sodium salt. We determined the critical aggregation concentration CSC of the amphiphils in water. From the figure and table data listed in table, there are two breakpoints of each curve corresponding to 2 CSC values. The TEM observation reveals that biliar aggregates as well as vesicular and ribbon-like structure. To observe the structural pattern of these amphiphil contains an amide linkage next to an aromatic entity with a long hydrophobic tail, in addition to polar head groups, which facilitates the gelation process. This is the schematic diagram of the gelation process.
The table represents the gelation behavior of the amphiphile in basic aqueous solution. Optical FSM and TEM techniques were confirmed the existence of nanofiber constituted the hydrogel of amphiphiles. The XRD data which reveals that the gel state the gelation molecules self-assembled in bilayer as well as in columnar square packing. Also, rheology studies support the gel network are mechanically stable and elastic in nature. CD technique also suggested the chirality presented in the gelation in aggregated structure. FTIR study recommended existence of mixture of alpha helix and beta helix. A proton NMR and DFT calculation supports the intermolecular hydrogen bonding. This is the schematic diagram of self-assembly manner of different isomeric solution. Applicability of gel were found efficient phase selective gelation, gelator, toxic dye removal, and reproducible nanomat formation. In summary, we have successfully synthesized 3 acyl amino group containing nicotinic acid derivatives and investigated their self-assembly behavior in aqueous solution and in basic aqueous medium. The gelator selectively gelates aqueous phase from oil water mixture. The amphiphile can also be utilized to remove toxic dye molecules such as rhodamine 6G, eosin Y, and rose bengal. The gelation aptitude of the amphiphiles in basic aqueous solution is applicable in tissue engineering as well as the formed hydrogels in basic aqueous solution successfully produce reproducible mat formation. The whole work was published in chemical paper 2020. Thank you.
Okay, so
Madhu, please continue.
Okay, thank you for your nice talk and a detailed study, I would say. And Professor Bagh has one question, so please, Professor Bagh, go ahead with the question.
So this is very nice work and very nice presentation. I would like to know, like, where from the chirality is coming. Like, do you have some chiral center in the molecule?
Chirality.
Now, do you have any chiral center in the molecule?
Yes, sir.
Could you please show the synthesis once again? I would like to see the spacer or the chiral center. Yeah. So these are chiral, is it?
Yes, sir.
Okay, fine. Very nice.
Okay, so any more question?
I have a small query, Madhu.
Yes, go ahead. Please.
Yeah, Siddhartha, just only one thing that you said that you have studied using NMR spectroscopy about the intermolecular hydrogen bonding formation.
Yes, sir.
But you are talking, these are the gelators. So how do they study the gel using NMR?
Sir, actually we started in solid sample in different solvent, DMSO-T6 and CDCl3.
So if you do this, so the gelator gel will be broken into a small molecule, right?
Yes.
Then how can you confirm the that intermolecular hydrogen bonding through NMR?
In DMSO solution, the amide bond is
okay, no problem. I got so no. Okay,
amide bond is 10.79 ppm, and in CDCl3 they shifted to the amide bond shifted to 9.22 ppm.
That is due to the DMSO polarity that can induce the basically hydrogen. So That is a solvent hydrogen bonding. So you cannot say that the intermolecular chelator hydrogen bonding.
There's also the DFT study supports the hydrogen bond.
DFT is just a
No,
calculation.
DFT will not give you the actual hydrogen bond information anyway. But the question is, I think Professor Mal is, I think, it's fantastic. The question is very valid, but you need to find out the answer for that. Actually, there is an answer.
Okay, so I'm done. Yeah, Madhu, please go ahead. We can go for the next presentation.
Okay, so thank you, Professor Mal. So should I call the next presenter, Professor Mal?
Yes. Thanks, Professor Parasuram. Yeah, sorry to interrupt you. Yeah.
No, you are right. You are right. I am sorry that I have interrupted you.
No, you're in the middle of the discussion. Okay, all right, no problem.
Okay, so let us then call Mr. Jagabandhu Ray. He's also from Midnapore College and working under Dr. Tridip Tripathi. So, Jagabandhu, can you please start your presentation?
Yes, sir. Am I audible to all, sir?
Yeah, you're audible. Can you please share your screen and presentation actually?
Is the slides are visible, sir?
Yeah, nicely. Can you please continue?
Okay, thank you, sir. Good morning, everybody. Myself, Jagabandhu Ray. Currently, I am doing my research work under Dr. Tridip Tripathi at Midnapore College. My topic is adsorptive removal of dimethoid and chloropyrifos pesticides from aqueous medium using dextrine graft poly 2-acryl amido 2-methyl propan sulfonic acid co-acrylic acid copolymer. So firstly, water is most essential ingredient for necessary activities of living organism. However, due to rapid development of unplanned urbanization, population expansion, and intense industrialization, various toxic elements comes into the aquatic system, such as various toxic metals, toxic dyes, and also various pesticides from the agriculture wastewater. So these effects on aquatic life and human health also. So now we should concern about the cleaning process of the wastewater before it's released into the aquatic system. Several techniques have been used, such as membrane filtration, chemical precipitation,
Some problem? Hello?
Yeah, looks like internet problem.
Are you there? Are you here?
I think he might have been disconnected.
Okay.
So
let's wait for 1 minute and then others will request the next speaker.
Yeah.
So Shravani is around? Ms. Shravani Saha from Viswanathi. Shravani? Prithidipa, are you here?
I am there. He's there.
Okay.
He's there.
So can we request Shravani to start it now, because Jagabandhu will come back again. We can accommodate him in the last or before after Shravani is let out. Madhu, is it okay?
Yeah, obviously. Yeah.
Yeah. So, yeah, please introduce her.
No, please continue. Yes, I'm just saying it. Okay.
Okay, then since there is a kind of disconnection, so now we can request Ms. Shravani Saha to start her presentation, and she is working with Prithidipa Sahu, Professor Prithidipa Sahu at Viswanathi Santiniketan. Shravani, please, if you could please share your slides. So basically, Shravani has also written, she has also some problem. So I think we can go for another next one. Is it okay, Madhu?
Yes, Professor Mal, please continue. I'm just here. Please continue with the remaining speaker. I'm just waiting here.
So, poster number 8, Mr. Subinoy Saha, are you around? Subinoy Jana.
Subinoy Jana. Subinoy Jana.
Yeah, Subinoy Jana, yes.
Am
So
I
are
audible?
you ready? Are you ready for your presentation so that we can basically
Yes,
continue? And okay, then requesting Subinoy Jana to start his presentation, and he is working with Professor Tridip Tripathi at Midnapore College Autonomous. Okay, so Subinoy, please. So this is poster number 8.
Sir, am I visible?
Yeah, you are audible and your slides are visible here.
Thank you, Chirantan Rasayan Sangsa, for giving me the opportunity of my research work, sharing the research work. Today my topic is adsorption of cationic and anionic dyes from their aqueous solution by pH responsive catiragram cross-linked polyacrylic acid co-N-vinyl hydrogel. Introduction. Water research. Water pollution. In recent year, water and environmental pollution is increased day by day with increased industrialization and civilization. In many industries such as food processing industry, rubber industry, textile, cosmetic, printing, use their synthetic dye for coloring their product. From their environmental point of view, removal of dye from wastewater is necessary before the disposal into aquatic environment. Various treatment process such as flocculation, adsorption, solvent use, but adsorption is most effective. Use to its simplicity and higher efficiency. The synthesis procedure for this dye, the pH sensitive hydrogel is synthesis initially. Sulfate anion free radical is produced by thermal decomposition of KPS, and this radical anion abstract from hydrogen from the hydroxyl group of the Kg backbone and form a macro radical by reacting with acrylic acid and N-vinyl imidazole. After that, macro radical form a three-dimensional cross-link with reacting with MBA. This is the characterization, FTR analysis and XRD. We studied characterize the hydrogel and two new peak at 1585 centimeter and 1648 centimeter assigned to CN and CC vibration of imidazole ring, and another peak at 1734 centimeter is due to CO group of the acetic acrylic acid group, which suggests that acrylic acid and N-vinyl imidazole is cross-linked with catiragram. And XRD study also support the formation of this hydrogel. And same analysis. The first same is polysaccharide catiragram, and then it cross-link, it form a irregular porous surface. After adsorption, surface become rough. This is the soiling studies of this hydrogel. The soiling studies show that two maxima it is observed from this study at pH at 4.5 and pH 9.0, which indicate that soiling properties is pH different, and PZC, that is 0 point charge of surface of the hydrogel. The surface charge less than PZC, less than pH less than PZC, surface become positive, and pH greater than PZC, surface become negative, and PZC of this hydrogel found to be 6. And the cationic anionic dye removal from aqueous solution, we studies mainly methyl violet and methylene blue, carmosine A and tratazine, and use this equation for removal, adsorption dynamics, effect of contact time, and temperature, pH, and concentration studies for removal of this dye cationic anionic from aqueous solution, and adsorption isotherm, Langmuir and Friendly's adsorption isotherm. We studied to find out the mechanism, and R-square value support that. Langmuir isotherm is fitted well with the experimental data. And kinetic study, we also study the kinetic field pseudo first order kinetics and pseudo second order, and from this R-square value we suggest that pseudo second order kinetics will be fitted well with this model. And desorption studies, Desorption and reusability is the important parameter for absorption to reduce its cost and make it suitable for industrial application. From this study, it is suggested that pH-sensitive hydrogel can be used repeatedly. This is the comparison from other reported adsorbent, and this adsorbent suggests that all dye are removed from this aqueous solution, but other reported adsorbent used only for 2 or 1. Conclusion: soiling study with water are carried out at different pH, 2 maxima, 4.5 and pH 9, and absorption dye is influenced by pH of the solution, and maximum absorption is found at pH 2 for TA, tratazine, and pH 3 for carmocine, and pH 7 for MB, and maximum absorption capacity found to be 331.5, 286.1, and 273.5, and 201.53 mg per gram for MB, MV, and CRTA respectively. And this developed hydrogen can be used efficiently and removal of the both cationic and dye at a particular pH from the solution. And this work will be published in colloid surface air, physiochem, and engineering aspect. Thank you for listening.
Okay, so thanks, Sunay. There are a lot of work you have shown. Very nice. And now I'd like to request audience to ask the question. Any questions from the audience? Okay, so I have one question basically.
Yes, sir.
So the swelling effect is showing at the pH maximum at 4.5 and maximum at 9.
2 maxima.
Yeah, 2 maximum. Okay. So why is it so? Means I'm not able to got the mechanistically. Can you please explain why exactly it's happening that why the swelling is maximum?
When pH become 1 to 4, the imidazole ring become protonated, and also in dimethylene acrylamide, that is cross-link, are also protonated. So repulsion will be occurred, and soil hydrogel pore will be increases. So soiling also increases. After that, it will be decreased due to protonation of the imidazole ring, and also acrylic acid will be deprotonated, that is dissociated. So weak interaction will be occur. For this reason, it will be decreases. After that, when pH will be increased, the only COOH group ionized, and also imidazole ring will be deprotonated. So COO group will be hydrogen bonded with the hydrogen water molecule to form a maxima.
Okay, thank you. I got the answers. Very nice. And any other questions? So if not, then we'll like to require. Okay, thank you once again for your excellent work. Now the next presenter probably if Shravani or Jagabandhu, if they are around, so I can request them. Okay, Shravani, please start your presentation. So Shravani is presenting. Her poster number is 7. Yeah.
Sir, myself Jagabandhu, I am my network was some problem is there, so I lost the connection. So
no problem, Jagabandhu. So let's one of the participants finish it. So since Shravani is around, so I'll request Shravani to finish it, and then Jagabandhu please join then afterwards. Okay.
Okay. Thank you, sir.
Okay, Shravani, please. Yeah.
Can I present my poster?
Yeah, please present here.
It is visible, sir?
Yeah, it's visible, but zoom it.
Is it okay, sir?
Okay. What's the title, please?
Yes, sir. At first, I would like to thank CRS for giving me such opportunity, and myself Shah from Vishwabharati University, and presently I am pursuing my PhD work under the supervision of Dr. Shah. Today, the topic of my poster is involvement of a unique pyridine-based chemosensor for the estimation of aluminum 3+ in rice field on cell imaging application.
We all know that nowadays aluminum toxicity is a major concern to all living organisms, such as the animal kingdom as well as the plant kingdom. Aluminum is widely exists in earth crust and it is widely used in many human activities like cooking utensils, manufacturing of cars and computers, infrastructural materials, municipal water treatment, and many more. An elevated level of aluminum in humans can cause several adverse health effects such as Alzheimer's disease, osteomalacia, Parkinson's disease, and even breast cancer. In the other scenario, aluminum toxicity has a pivotal consequences in the plant growth, such as inhibition of root cell growth and division, nutrient imbalances, production of reactive oxygen species, and which finally results in reduced plant production. So, it is desirable to introduce a simple portable tool to monitor the aluminum toxicity level. In our present work, we have developed.
Prosenjit, could you switch off your microphone? Prosenjit, switch off your microphone. It is disturbing. Okay.
Yes, sir. So, for the sake of health and environmental issues, we have developed a pyrene-based chemosensor which can selectively recognize and quantify aluminum 3+ efficiently in the roots and stems of the rice plant through turn-on signaling. Here, this is the crystal structure of our synthesized probe PTI, which is pyrene-based, and this is the schematic representation of the mechanism through which our probe PTI binds with the aluminum 3+ ion. To know more about the interaction mechanism, we also perform the UV visible and fluorescent titration spectrum studies. Here we can see that in the fluorescent titration studies, the probe PTI is almost non-fluorescent when excited at 350 nanometer. But upon addition of incremental doses of aluminum 3+ ion makes 39-fold fluorescence enhancement at 431 nanometer. Hence the fluorescent color change. We also perform the selectivity experiments with several biorelevant metal ions, and here we can see that only aluminum 3+ ion can change the non-fluorescent nature of the probe PTI and shows vivid blue fluorescence color. To know more about the interaction pattern, we also perform the proton and 13C NMR titration studies. This is proton NMR titration studies, and the shifting of the peaks confirms the obvious interaction between the probe and the analyte. Now, we also calculate the time-dependent density functional theory, and this is the HOMO and LUMO distribution of the PTI and the PTI aluminum 3+ complex. And this is the confocal microscopic images of the root and stem taken from the rice sapling. Here we can see that our synthesized probe PTI can efficiently recognize the aluminum 3+ ion within the cells, root and stem cells taken from the rice sapling. And we successfully quantify the concentration of aluminum 3+ ion in the rice plant root extract. And this schematic representation shows the preparation of the root extract by comparing with the standard fluorescence curve. We obtain the concentration of aluminum 3+ ion in the root extract as 0.16 micromolar. So, to conclude, we can say that our synthesized, we have developed a simple probe PTI based on pyrene, which can recognize aluminum 3+ through turn-on signaling with high affinity and selectivity. It also successfully employed to estimate the concentration of aluminum 3+ in rice plant root extract along with the cell imaging in the root and stems of the rice sapling, and sufficiently lower detection limit and the applicability in the live cell makes it a potent tool which we need. So this work is published in Chemistry Select in 2019. That is all from my side.
So is it over?
Yes, sir.
Okay. So thank you. The session is open for discussion. I'll like to request the audience if any questions you can ask. Okay. So nice work, Savani. So any questions so that I can ask one then? Madhu, can I ask one? All right.
Please, yeah,
Okay.
please.
So yeah, Savani, one thing I have just so you have shown that pyridine attached to one of the thiophene ring. Okay.
Yes, sir.
So pyridine amide attached to thiophene ring. In that case, sir, you have shown the arrow of ICT that is from the thiophene ring to the pyridine ring. But I think it should be opposite because nitrogen is more basically it can donate the electron. So the pyridine ring overall it will be more electron rich than the thiophene ring. Okay. So the is it? I don't know because I am not expert. So I just curious about the arrow of the ICT that on you are showing. So it should be reverse. Am I right?
Okay, sir. But from the density constant theory calculation, we can see that there is a homogeneous distribution.
No. See, I don't know because I am an organic chemist. From our understanding, so I'm just looking at that. Okay, that how the ICT arrow is given. I think it should be reversed. So that is my query. If you can basically tell that what exactly is happening, I'll be happy to know that because I don't understand any density functional theory.
From the density calculation, we can see that the HOMO and the LUMO distribution, HOMO and LUMO congested over, I mean distributed all over the molecule here in PTI. But when PTI complexed with aluminium 3+, the HOMO and LUMO congested over the pyridine moiety.
No, I think that is not the point I asked for. So I just curious about the arrow pushing direction.
And here we can see that the ICT, I mean the intermolecular charge transfer inhibited, and this is empty. I mean HOMO and LUMO orbital contribution here is
Okay, got it. So no problem. So any other questions? Okay, so if no more questions, then I would like to thank Shravani for excellent presentation. And the next speaker, I think Jagabandhu is waiting because some problem was there. So I would like to request Jagabandhu to start again.
Yes, sir. Am I audible, sir?
Yes. Please start.
Yes. Is the slides are visible, sir?
Yes, you are. Yes.
Okay, sir. So, water is the most essential ingredient for necessary activities of living organism. However, due to rapid development of unplanned urbanization, population expansion, and intense industrialization, various toxic elements comes into the aquatic system, such as various toxic metals from different metallurgical industries and dyes from textile printing and pen industries, and various pesticides from agricultural wastewater. So these effects on aquatic life and human health. Now we should concern about the cleaning process of the wastewater before it's released into the aquatic system. Several techniques have been used: membrane filtration, chemical precipitation, electrodeposition, reverse osmosis, etc. Now among them, adsorption study we have chosen because this is very simple and highly efficient and economically feasible. So we choose the adsorption process. Here is the synthesis and mechanistic path for the graph copolymerization formation. So first of all, potassium peroxide disulfide K2S2O8 is used as a free radical initiator, which decomposes at 70 degrees Celsius to give sulfur radical anion, where we have used AMPS and acrylic acid monomer to propagate the chain. After termination step, we got the graph copolymer. Then the polymer is synthesized, characterized by these techniques. In the proton NMR spectra of the dextrin, we got peaks from 3.46 to 5.27 ppm, which corresponds to protons of the dextrin polysaccharide. And then in the polymer, new peaks generated at 1.49 and 2.70 ppm corresponds to methyl proton and methane proton respectively. And then in the FTI spectra, polymer 2 bands at 1657 and 1560 were assigned for CO splashing and NH bending respectively. These two, the two sharp bands at 1042 and 630 semi-inverse are identified for SO and CS stretching vibration. After pesticide adsorption, new peaks for P double bond S and PO stretching frequency are found at 746 and 972 semi-inverse for dimethoate loaded polymer, and 854 and 1018 semi-inverse for chloropyrifos loaded polymer. For chloropyrifos, two other extra peaks at 692 and 1412 semi-inverse are appeared due to CCL stretching and skeleton vibration of the pyridine ring respectively. Other polymeric peaks are shifted. So that means the chloropyrifos and dimethoids are adsorbed onto the polymeric surface. And then XRD plots, we get a broad peak for all the compounds with changing the theta value only. So that indicates that amorphous nature of the compound. Then same image, we got a smooth homogeneous surface for the dextrin, and then the surface becomes porous and heterogeneous for the graph copolymer in the figure B. So that which is ideal for the adsorption process. And then after adsorption, again the surface becomes quite smooth. And in the typical batch mode adsorption study, we have taken the pesticide solution of known concentration, and then polymer solution was added to it. And after certain time interval, 3 to 4 ml of pesticide solution was taken out, and UV spectra was measured at their corresponding lambda max value. And this equation we have used to calculate the percentage of removal efficiency. And then different different operating variables have been studied to find out the optimum condition. From the contact time studies, we got that initially adsorption was greatly increased and equilibrium was reached at 70-minute time. And from temperature study, we found that maximum adsorption at 55 degrees Celsius. Beyond this temperature, adsorption decreases. So then if we change the initial pesticide concentration, we see that adsorption efficiency decreases with increasing concentration. And then maximum adsorption efficiency was observed at pH 4, which is close to PZC value. That means at this pH, the polymer chain contain the undissociated COOH and SO3H group. The electron donating sulfur, nitrogen, oxygen, and chlorine of the pesticides can form strong hydrogen bonding with the polymer. And then electrostatic attraction may also occur with the partially positive phosphorus atom. These are shown here. And that means hydrogen bonding and electrostatic attraction are the mainly responsible for the adsorption. 4 different kind of kinetic model have been studied. Among them, good fitting is observed for the pseudo second order model, whose correlation coefficient value is close to 1, which is shown in the table. That indicates that adsorption depends on the amount of pesticide adsorbed on the surface of the polymer, as well as on the amount of amount adsorbed at equilibrium. Again, among all these adsorption isotherm model, both the Langmuir and DR isotherm model fitted well. It is proposed that monolayer coverage of the pesticides over the homogeneous surface of the adsorbent. And for dimethoate, maximum adsorption capacity is found to be 172, and for chloropyrifos it is about to 108, which is shown in the table. And we have compared the maximum adsorption capacity value with other reported adsorbents. We got good results. And now in the desorption study, we have used 3 different stripping solutions for the desorption study, pH 3, 7, and 8. Dimethoate and chloropyrifos are mostly desorbed at pH 8, that means in basic medium. 3 adsorption-desorption cycle are studied and we have seen that about 70 to 80% of the polymer is recovered after 3rd cycle. And here is the conclusion of my study. Dextrin graph poly AMPS co-acrylic acid graph copolymer is successfully synthesized by free radical polymerization technique. And we have seen that it is pH sensitive, and the maximum adsorption is observed at pH 4 for both the pesticide. The graph copolymer has excellent pH adsorption efficiency with high Qmax value, and the adsorption is controlled by pseudo second order kinetic model, and adsorption isotherm follow Langmuir and DR model both. And the desorption experiment suggests that graph copolymer can be used repeatedly for several times for the adsorption of pesticides. There are some references are there. Thank you for your kind attention.
Okay, thank you for your nice talk and a lot of work. And any questions, please, from the audience? Okay, so if no more questions, I'm thanking Jagabandhu again. And I'd like to request Madhu to introduce the next speaker.
Thank you, sir. Thank you, sir.
Okay. Okay. Thank you, Professor Mal. So now next speaker is Hillol Khatua. He is also working CBMR Lucknow under the guidance of Professor Buddhadev Chattopadhyay. He will be talking on the iron 2 based metallo-radical activation chemistry. So let us directly go to the talk. Hillol, can you start your presentation? Are you there?
Yes, sir. Yes, sir.
Yeah, please
So,
continue.
Jawaharlal, can you please stop your slides so that I can share my.
You start your sharing, it will be automatically going.
Okay. Sir.
And enlarge, and as
Yes,
everybody
yes.
doing, and start.
So good morning to all of you. I am Hillol Khatua from Center of Biomedical Research, Lucknow, and I'm working under Dr. Buddhadev Chattopadhyay. Today I will be talking about iron catalyzed metallo-radical activation, switching from traditional click chemistry to denitrogenative amylation. So in this work, as you can see, we used tetrazole as a starting material, and as I have shown over here, the tetrazole remains in close open equilibrium. Our idea was to generate nitrine from this open form of tetrazole, and we wanted to trap that nitrine using alkyne to have imidazopyridine derivative. What are the major challenges we faced during this project? So there are two major challenges. One is I have shown over here the favorable azide alkyne cycloaddition product, which lead to triazole synthesis. This path we have to suppress in that time. So our another challenges was to generate nitrine from tetrazole. Now these two challenges were there in during the project. So in the previous, if you see the Weintraub group working on this field, he generated nitrine from this tetrazole using FVP process. That nitrine undergoes uncontrolled nitrine rearrangement to end up with various nitrogen heterocyclic compounds depending on the substitution, as I have shown over here. So our idea was to generate a stable nitrine employing transition metal, and we wanted to generate, we wanted to trap that nitrine using alkyne to have imidazopyridine. If we could do that, that will be the third dimension for the application of tetrazole. So after extensive screening with various transition metal, we found that iron porphyrin along with zinc dust effectively generate that iron nitrine radical intermediate, and that iron nitrine intermediate, radical intermediate reacted with alkyne to have this nitrogen heterocyclic compound imidazopyridine. With this optimized result, we screened a lot of alkyne as well as substitution at the pyridine ring of tetrazole, and we synthesized library of imidazopyridine derivatives as I have shown few over here. Also, we synthesized two important biologically active compounds. One is TP3003 and another is GABA agonist using our developed method. So after lots of experimental screening and experimental studies, we end up with a possible mechanism where iron TPPCl reduced to iron 2 TPP using zinc dust in the reaction medium, and that iron 2 TPP is the active catalyst for our reaction. That iron TPP coordinates with the open form of tetrazole to form this intermediate B. That intermediate B, upon abolition of nitrogen gas, generates that intermediate C, that is the iron nitrine radical intermediate. We identified that iron nitrine radical intermediate using EPR spectroscopy. After that, we trapped this nitrine using alkyne, and that reaction of intermediate C with the alkyne generates intermediate D. That intermediate D undergoes radical cyclization to have intermediate E. That intermediate E, upon generation of our desired product, produces active catalyst A, and the catalytic cycle regenerates. So at the end, I would say that we have developed a nice method using iron porphyrin catalyst for intramolecular denitrogenative transannulation, which completely overrides the traditional click reaction between azide and alkyne to have successful annulation reaction. And I am happy to share that we have published this work in Invocation in 2019. And at last, I say that I am very much grateful to CSIR New Delhi and DST ACRB for the funding. And I also thank my supervisor and the director of CBMR. And at last, I also want to thank the organizer of this RASAYAN Samstha for giving me the opportunity to present my work in this platform. Thank you so much.
Thank you, Hillol. It is yet another beautiful demonstration of radical chemistry, what your group is doing. Really fantastic work. So, is there any questions? Is there any question from the audience? So, no question. So, Hillol, I have one query here only. When it undergo decomposition, you get the azide, right? It is in equilibrium. So my question is, if you take 2-chloropyridine and react with sodium azide, will it give you this azide complex? Is such precedent there?
Made from 2-chloropyridine, and we use we use 2-chloropyridine for the synthesis of the tetrazole.
So how you are doing that? Using sodium azide or?
It's a sodium azide in acetic acid, water, and ethanol mixture of solvent. We heated it to 120 degrees Celsius, 24 hours to get this tetrazole compound.
Okay. So it is always in mixture or it is only in equilibrium in the reaction mixture. But
actually, sir, it depends on the temperature. When at room temperature, when we took the NMR, it is only in the closed form, and when we heat it to higher temperature, then it remains in the closed open equilibrium.
And how much explosive is this molecule? Because so many nitrogens are there.
Very much stable, sir. We heated this reaction up to 150 degree, even at 180 degree also. The material remains intact in the reaction medium without any catalyst.
Okay, Hillol. Thank you very much. It's really excellent work. I have already gone through last year. So it's really nice work. Thank you for your nice presentation. Okay, so Professor Mal, so last speaker waiting and
yeah, please Madhu, you go ahead, no problem. So
okay, I think last speaker will be for Soikat Kumar Paja, and he is from Professor Bagh Research Group only. So he will be presenting on terpenoids, nanoentities, and molecular cell assemble. Okay, on this. So I request Mr. Paja to start your presentation.
Good afternoon, sir.
Can you share your screen?
Sir, is it visible?
Not yet. Yes, now okay. Coming.
It's coming.
Yeah.
Sir, is it visible?
Yes.
Okay, good afternoon, sir. Myself, Soikat Kumar Panja, working under Professor Rajagopal Bagh. Today I am presenting poster on terpenoids, nanoentities, and the molecular cell assemblies. Plants are the rich source of innumerable number of phytochemicals. These have tremendous significance for the development of sustainable society. During last 2 decades, several terpenoid molecules have been isolated from different parts from different medicinal plants in our laboratory. Terpenoids are multiple of C5 isoprene unit, from monoterpenoid to higher terpenoid like triterpenoid, diterpenoid, tetraterpenoid. They are biosynthesized in plants enzymatic pathway. In organic compound, carbon, nitrogen, oxygen, and hydrogen are the common elements, and they have the diameter in sub-nanometer range. Computational studies and DFT calculation study carried out taking hundreds of number of molecules revealed all the terpenoids have the length in the range 1 to 4 nanometer, and they have several functional groups and large lipophilic moiety, so they can be termed as functional molecular nanos.
They can spontaneously self-assemble in liquid, and this is the schematic representation of self-assembly of the molecular functional nanodes, building different architectures like vesicles, spheres, seeds, helix, petals, flowers, etc. These are the examples of the molecules yielding vesicles. These are the diterpenoids and the studied triterpenoid. They spontaneously self-assemble to form vesicles and gel. And these are the optical and electronic microscopic images of the vesicles and tubes formed by the spontaneous self-assemblies of this molecule. As the vesicles have some void space inside, they can easily entrap different fluorophore, including anti-cancer drug doxorubicin, and the epifluorescence microscopy images clearly indicates they are capable to entrap various fluorophores, including anti-cancer drug doxorubicin. These are some of the molecules studied in our laboratory, and these spontaneously self-assemble to form gel and flower-like architecture by the formation of 1D fiber, fibrillar network, and petal. And the schematic representation of self-assembly has been shown here. These molecules spontaneously self-assemble to form various architectures like fiber-like network and flower-like network, flower-like architecture, and petals. As these microstructures has high surface area and porous nature, they could easily entrap toxic micropollutant and can remove them from water. And these gels are also used for the release of different anti-cancer drugs at physiological pHs. In a summary, we have showed that terpenoids have the length in nanometric dimension and have large lipophilic backbone and several functional groups, makes them an unique amphiphile for studying their self-assembly property, and they can form different architectures and they could be utilized for different purposes. In conclusion, we have shown that terpenoids could be utilized as renewable molecular functional nanodes. They can spontaneously self-assemble in liquid, yielding nano to micro-sized architectures like vesicles, fibers, tubes, flowers, etc. The self-assembly has been utilized for the entrapment and release of fluorophores, including anti-cancer drug doxorubicin, removal of toxic micropollutants, generation of hybrid materials, etc., paving the way for their future applications in nanoscience, biotechnology, environmental remediation, recyclable catalyst, etc. Thank you, sir.
Thank you, Shekhar, for your nice presentation. So now I ask audience for if they have any questions.
I have a question, basically, the last speaker. So,
yes, sir.
So you are saying that it's a spontaneous self-assembly, but it looks like that the same material is giving a different morphology. So what is the driving force?
So actually, self-assembly, we have started the driving force via FTIR.
No, I'm not asking that. I'm asking that it's the same molecule giving a different morphology, whatever the structures you have shown in the schematic, like rod, like nanofiber, 3D architecture, sphere. Then what is the stimuli? What is the stimuli basically to get a particular structure?
Actually, we have observed that the molecules having dihydroxy group, as I am showing here, these are the class of molecules which form spontaneously flowers and fibrillar network, and
these are the molecules having hydroxyl and carboxyl groups at the two opposite ends spontaneously forms.
Okay. So you are saying the different molecule, that functional groups are different, and then they are giving a different structure.
Yes, sir.
It's not the same molecule giving a different structure in terms of a different stimuli or something like that, right? Okay, got your point. Yeah, I'm done.
Yes, sir.
Okay, thank you, Mr. Paja, for your nice presentation. I think this is the last talk. I hope we did not miss anything, right, Professor Mal?
Yeah, hope so. And the good thanks, Madhu, for.
So I now request Professor Mal to conclude the poster session.
Pardon?
Yeah, okay. I request you to conclude the session.
Okay, thanks, Madhu. And now I'd like to thank all of you for being here and listening to the talks. And of course, Madhu has done the excellent job. And of course, finally, that Professor Pajakumar Bagh, that who is the organizer and the president of the CRS. So thanking him again, and hopefully the students, those who have done well. So hopefully, I would like to see that you are doing much better in near future. All right.
Okay. Thank you. Now,
Mr. Pradhan, Mr. Pradhan.
Yeah.
Pradhan.
Yes, sir. Okay, so thank you all the participants for their insightful research presentation. Now we are actually at the end of our session. We are going to begin our valedictory session, and this will be chaired by Dr. Jagannath Pal. So, Dr. Pal, over to you.
Thank you. Good afternoon, everybody. On behalf of Midnapore City College, I take this opportunity to propose the chairperson, and also I would like to here present and to both of thanks to these who have directly and indirectly contributed to this sixth one-day webinar on science beyond boundary, invention, discovery, innovation, and society, RASAYAN 10, organized by RASAYAN team. I would like to thank Professor Sonku Mallik and Dr. Silva Kumar Nastan and Dr. Abhijit Mondal for enlightening us with their knowledge. Today's webinar was full of knowledge and interesting things, and we are also inspired by your great words. And also I would like to thank Chairperson Professor Jayanta Kumar Roy, Professor P. Jayasankar, and Professor Madhu Sudan Maji, and Professor Prasanjit Mal. And also I would like to pay some thanks to the organizing committee for their unflinching support and coordination. And our heartfelt and thanks to all students for their active participation. And I would like to thank Professor Bujagopal Bagh, convener of this webinar, for inviting Midnapore City College for conducting in this sixth one-day webinar. Thank you, all of you. And now we move to the end of to this webinar. And now I would like to request the Secretary CRS, Dr. Abir Chandan Barai, for concluding remarks of today's webinar. Thank you.
Thank you very much. Good afternoon, good noon all. Honorable guest, Professor Sudipto Chakraborty, Principal, Midnapore City College, and also the co-host of RASAYAN 10, Honorable former CRS Medal Awardee, present here today as guests to grace the occasion. The speakers in RASAYAN 10, Professor Sonku Mallik, Dr. Silva Kumar, Dr. Abhijit Mondal, and the chairpersons, Professor Jayanta Kumar Ray, Professor P. Jayasankar, Professor Prasanjit Mal, Professor Madhusudan Maji, Dr. Jagannath Pal, Mr. Prashanto Pradhan, faculty members, research scholars, masters, and graduate students from various institutions present here today. Ladies and gentlemen, it's my proud privilege to have the honor to propose a vote of thanks on this occasion. The sixth one-day webinar entitled Science Beyond Boundary, Invention, Discovery, Innovation, and Society, RASAYAN 10, jointly organized by the Chirantan Rasayan Sanstha and Midnapore City College. I, on behalf of Chirantan Rasayan Sanstha and the entire fraternity gathered here today, including the media persons, and on my own behalf, extend a very hearty vote of thanks to our honorable speakers for kindly responding to our invitation in spite of their extremely busy schedule. I also thank Professor Brajagopal Bag, the founder president of Chirantan Rasayan Sangstha and convener of this webinar. I also thank honorable guest Professor Sudipto Chakraborty, Principal, Midnapore City College, and also the co-host of Rasayan 10. We are thankful to former CRS Medal awardee to be present in this webinar and for their encouragements. We are also thankful to Dr. Prithideepa Sahu, Vishwabharati University, Dr. Abhishek Ghosh, and Dr. Jagannath Pal, Mr. Prashanto Pradhan, and Mr. Sumon of Midnapore City College, who played a very active role and was highly instrumental in organizing this webinar. We are also thankful to all faculty members, supporting staff, students, and research scholars, media persons to be present here to make this event a success. Last but not the least, we thank the Google Meet platform, YouTube, for providing such an opportunity to assemble in this manner. Thank you very much to you all.
Thank you. Thank you. Thanks a lot.
Okay. Thank you. Bye bye, everybody.
Thanks.
Okay, bye. Yeah.
Well done.
Bye bye.